There's still nothing here.
It's just the black screen.
Oh, it's just started. Yes. Yes. Okay, sorry about that interruption. There was a power issue that led to an internet issue, but we're back online. So thanks for your patience, Alana, and thanks to the other speakers who are waiting. I pass back over to you for your presentation on shrimp fisheries. Good luck. So hello, my name is Alana Duffy, and I'll be presenting you my master's thesis under the title Innovation in Sustainable Fishery for a Brighter Future, Testing Odor Attraction in Cranon for pot fishing. So, as some of you may know, the fishing industry today is facing a couple of different pressures, one of which is the increase of offshore development, for example, to create renewable energy under the form of offshore wind farms, which often are quite fragile and not compatible with most current fishing methods, reducing the areas allocated for fishing. Another issue, for example, is the most common fishing method is bottom trawling, but it is also the most unsustainable. As the gear is constantly in movement, it uses a lot of fuel and releases therefore a lot of carbon. Also, it has a lot of bottom contact and is very damaging for the ecosystem. For these reasons, we would like to find a more sustainable way of fishing and also a way that could be compatible with multi-use areas like offshore wind farms. Here you have the illustration of a bottom trawl, so you have a beam to hold the net open and then a chain to drag and capture. The prey, this one specifically, is meant to target brown shrimps. So an option for an alternative fishing would be pot fishing, which exists today, where we have cages to allow different species to enter, but they're usually baited, and then they're left at the bottom of the seafloor and collected a few days later. Yes, so they are more sustainable because there's no movement, they consume less fuel. Also, the bottom contact is less damaging as they're simply placed on the bottom and not dragged along it. And because the prey is kept alive, the bycatch release rate is quite high. Survival rate is quite high. Also, because there's no dragging, there's no risk of damaging the cables of offshore development. And its main issue today, though, is that it's not very efficient for quite a few commercially interesting species. But there hasn't been so much study yet to try and make it more efficient, so that's what we're aiming to look at. We decided to look at smell as an attractant for a couple of reasons. There was already a few literature on light and very little on odors. In seas like the North Sea, the water is very turbid and light doesn't travel very far, whilst odors can go much further. Finally, often marine species have a very strong olfactory sense, so it could be a good option. We picked Cranon as our target species, or commonly known as the brown shrimp, as it is the third most profitable species in the North Sea. It's also a fishing industry that's particularly damaging because the net is very small and also impacts juveniles of other species that are of commercial interest. And so far we haven't found any literature talking about fishing Cranon pots. So we started by sampling shrimps. So we did that in two ways. We had two campaigns in black on the map here. Here you have a picture of the trawl that was used in the campaigns. Obviously it's at the surface so that you can see it with the picture. But it would have been at the bottom, trawling along the bottoms. We also had 4 samplings at the coast with a hand trawl that was dragged on beaches. The shrimps were kept in tanks with some sand at the bottom, as this is a burrowing species. Those tanks, they were the same for holding and just preserving the shrimp, and then the experimental ones. This way the conditions would be the same. And you can see the light is red because often the experiment would take place during the night cycle, and we assume they can see red light. So we had two experimental designs. This first one was actually in a different setup than the one I just showed because the tank was much bigger and had to be in a different room. Again, a bit of sediment at the bottom. There was about 500 shrimps in the tank, and we put 5 different pots at different places, and they were baited, left for 24 hours, and then we took out the pots and then froze the shrimp they had captured. The idea was to see if, in function of the different pots, maybe some of them were more efficient at catching the shrimps. So we counted the number of shrimp they had caught, weighed them, and then measured their size. So that was 3 response variable, and for analysis we did a one-way ANOVA with the one response variable in function of either the pot or the position to see if that was actually what was going on. So here are the results. So you have in function of pots and position, here you have number of shrimp, total mass, and average size. So unfortunately we only did 4 replicates because then the big tank was no longer available to us, and so we didn't get any significant results. But it is still interesting to note that pot 4 had a much lower number of shrimp caught and lower mass, but on average it seems like the size was bigger. So why didn't we get significant results? Well, probably because we had very few replicates. So if we had more time and access to that tank, we would like to do more. We did see that pot 4 seemed to be more size specific, and in looking at the characteristic of the pots, by the way, this is pot 4, it had the biggest mesh size. There was one with a bigger one, but it was diamond shaped, and one side was bigger and the other smaller. So that's interesting. And also, the reality is none of these pots are actually made for Prion because there's none commercially available yet. So I think this is interesting, but a lot more research needs to be done to figure out what makes a pot efficient in catching Prion. Second experimental design, the main one was this one, so in the tanks I had previously shown, where we put this divider in. The center section had the bottom of it cut out here in the graphic, and then there were escapes from the center as these funnels. They were meant to imitate like a pot entrance, where it was easy to go this way, but then because it's hard to go the other way. So we initially put 50 shrimps in the center with the funnels closed, waited for acclimatization, and then opened the funnels. In the 4 compartments, we put different treatments. And the assumption was that shrimps from the center would exit via the sides and not be able to go back in. And then after the experiment had run, we would close the funnels and then take out the shrimps and count them. And the idea was that the compartment with the most shrimp would be the most attracted to. Here you have a picture of it in action underwater. It's light, obviously, because if I had used Camel, you wouldn't be able to see the treatments. So for analysis, we used a general linear mixed model with the number of caught shrimp per treatment, and then we use an offset variable as the number of initial shrimp, as we aimed for 50, but sometimes this was miscounted, and a few died during the experiment time. And also we accounted for random events like the experiment number, or maybe the compartment, because you can see the water was coming one side, coming out another. That could have played. Oh, also, treatments were, between every replicate, switched around randomly. To kind of have an idea of where the odor was spread, we use a dye, methylene blue. It should have the same molecular weight as the odors we used. Unfortunately, it is toxic, so we didn't try it in the same exact setup. This was separate from the circulatory system, and there were no shrimps in it. So maybe the spread wasn't exactly what happened during the experiment. So we did 2 sets of treatments for this experiment. The first one had negative control, and then 3 exotic odors, and there were no significant results. Second set of experiments had a negative control, a positive control under the form of a whiting tail. And then here is the same exotic odor as here, because at some point it seemed to have the highest, but it didn't anyway. And then herring here is a synthetic fish smell that is sold for nature fishing as bait. Again, no significant results. So why didn't we get the results we were expecting? Well, there's a couple of reasons. We mostly focus on exotic odors, but this was because in the lab I was working at, they had done some tests for feeding flatfish, and it seemed to be working, but obviously salmon is a very different species, very distantly related, so they might not react the same. Second of all, as I said, we're not completely sure the odor reached the center. It's very hard to assess where odors circulate. Third of all, the experiment was in a circulatory system where the water was filtered, and the assumption was that the filter would get rid of the odor and then go back into the system. If that was not the case, then the replicates were not really useful, because the second we had done one first experiment, then the water was contaminated with all treatments. And then finally, there were very few shrimp this year, so we tried to preserve them as much to reuse for maybe other experiment. So we only counted abundance and not mass or size or any of that. So that would imply killing them. So what would we recommend for future research? Well, antenna actinography method is a technique developed to assess the olfactory response of insects, but today has started to be used in marine crustaceans. So we could use this to try and determine which odors can they actually perceive, and then do some lab experiments similar to what we did, or maybe simpler with just one treatment at a time to see whether or not they're attracted to that odor. And then finally, we would do experiments in the field, maybe using combined attractants like smell and light, and comparing that to traditional bait and see if we can use that. That's it.
Lovely.
Well done, Alana. Nice presentation. So does any of the audience first have any questions? Yeah.
Hi. Thank you so much for your presentation. I'm not sure if you mentioned this, but I was just wondering whether there was any literature or any understanding that the specific species you used in your study has sort of an olfactory system that's sort of developed, or like, is response to certain odors. Because, I mean, you mentioned that you used exotic odors such as strawberry, banana, and things like that, but are those scents that crustaceans
initially, the design was supposed to be with flatfish, and we had already kind of tried that initially, but then we weren't able to get the fish, so we switched to shrimp. I mean, they do normally get attracted with like actual live bait that they smell, so they can smell to some extent. Yeah, but the problem with live bait is that obviously you have to fish it, so again, that's not always sustainable, and it's a cost. And so the idea was to try and see if we can replicate that with something synthetic we could create from that.
Yeah, another question.
Just a very quick follow-up question. What is the traditional bait for the shrimp?
Well, it's like a piece of fish or like food, actual food they would eat.
Okay, thanks. To the jury members, I don't know who wants to ask first. Who wants to? Yeah.
Okay, so Elena, just to understand a little bit better, you said that in your first experiment that the position mattered more than the cost. Do you want to elaborate a little bit on that?
Yes, so none of this is significant, but if we took alpha 10% instead of 5, position was actually significant. You can see that E actually performed differently.
And why do you think that could be the case?
It could be where the water was arriving. So here you would have had the door, more light, and then here you had a water input, and then this was the position that seemed to capture most shrimp.
Okay. And then, yeah, so in your second experiment, you had different odors.
Yes. So it was. Oh no. Sorry. Yeah. So 4 experiments at a time with always a negative control, and then one time, well, one set of replicates was 3 exotic smells, and then the other one was negative, positive, and then the synthetic fish and the strawberry.
Yeah. Like, I don't understand why the exotic smell would be something that.
Yeah, again, initially it was tried with, to promote feeding behavior in flatfish, and it seemed to be working. And the project was initially started, I was supposed to work on. So
Because it's not something common in the
yeah,
environment.
no, I
so
agree.
they wouldn't associate it with food, I suppose.
But then we couldn't get the fish, and I was told to swap to Crown.
Understand. And then just lastly, so for how long would the experiments have lasted?
It was 16 hours.
16.
16 hours for one replicate, yes.
You had 4 replicates?
No, I had 4 replicates for this one. For this, I had 15 replicates, something like that. I can't quite remember. But yes, because they were long, and also I was sharing the divider with somebody else. I didn't have a lot of time to do lots of replicates or try different combinations of treatments.
And were you using the same animals for all of the?
No, so we had. So some of these tanks just were holding the shrimps we had collected, and then we had the divider in one of these tanks, and we would collect 50, put them in the center, and then once they'd been used, put them in a different tank for other experiments that were unrelated.
Okay. Thanks very much. Thank you.
Well, Hannah, I've got a few questions. I think that the way you designed the experiment was almost impossible to see very clear results, you know.
I didn't choose the design.
The first experiment,
Yeah.
you rotated the position of the smells in the 4 replicates.
Yes. So yeah.
So in fact, if you look at position, the type of not that one, the other one.
Oh, here. Here, no.
There's no smells, but
no
anyway,
smells.
you did it 4 times, and the
Yeah.
types of pots that were different, which actually is something that should be shown here. These were different types of pots.
Yeah, all different pots.
Okay, you did this 4 times. So
4
if
times.
you move the position
Yeah.
of the different pots, you have no replicates, because in the crossing of the pot and the place in the tank where the water flow is different, you have only one
No,
observation.
I agree.
So it would be very hard to see
I agree.
any effects.
Yes. No, unfortunately, the tank we didn't have any
Using the
more
same
replicates.
kind of pot, the most likely pot, only one probably would be better, because then you would have some replications. Anyway, another thing that I think is important when you present this, you put the size of the tank, because we have no idea if this is 1 meter, 5 meters. You know, you should have the dimensions, both the pots and the
Okay, yes,
the
I have
tank.
put it in the thesis,
Okay.
but I didn't
Now,
in the slides.
What I'd like to ask you is, I'm sure you did a literature review,
Yeah.
or what were the successful shrimp fisheries with pots that you can use as an example for this one?
There was not a lot. This is relatively new. I did find, I think, 1 or 2 articles that did. It wasn't necessarily the aim, but they didn't manage.
Commercial fishing for shrimp.
Commercial fishing for
Yeah.
shrimp with pots.
Why do you think that is? Because it's very hard to
Yeah.
catch shrimp with pots.
Yeah.
You know, I can tell you I've tried a lot without success. So you have a very difficult
Yeah.
issue, okay? And starting on a very difficult issue, doing a lot of things at the same time, types of smell, types of pots, it's very hard. My suggestion is the experiment should be done by stages with a lot simpler questions in each stage of the experiment. I'm sure that you did what the lab was doing, and this is not at all a responsibility, but my advice is that if you go on this research in the future, you have to be
Yes.
more humble in the objectives of each step of the experiments.
Yes.
Okay. All right, so that's it.
Thanks, Margareta. Urtzi?
Yes, okay. Thank you for the presentation, and if I well understood, the plan was to make it with fish.
Initially, yes.
And why is the reason? Because ethical issues?
I think
Obtain the
campaign that was supposed to collect the shrimp, for some logistical reason, they didn't get the permission.
I was imagining that.
Yes, so yeah, they didn't get permission.
The permission for the experiment and for fish or both.
To collect the fish with the ship.
Okay. So perhaps that's why you have, as my colleague was commenting, that different, just to have some results.
Yeah.
That was the point because of those mistakes or
No, so
not mistakes, but
the trial with the exotic odors had nothing to do with this. It was for aquaculture to promote feeding in flatfish and putting some synthetic banana smell on the food actually made them eat more. So they thought that maybe we could also use that to attract them.
and the question was if the idea is to go to the field.
Yeah.
Shouldn't be easier maybe go to the field directly and try? And if you catch more, boats, and if not,
Well, going to the field is expensive and takes a lot of resources. So they wanted to have an idea of what works before they went in the field.
it's expensive, but I mean,
If I didn't really.
the fisheries, it shouldn't be that complicated. I don't know if it's more complicated or not than this, but
No, I understand what you're saying. No, I was told to use lab experiments to kind of prep the field experiments.
because when you start doing the lab experiments, it's supposed that you have under control situation, but as you can see, sometimes it's not that control as we're supposed to. But this happens, maybe. Okay.
It's one aspect I forgot. 16 hours is a lot.
Yes, because weren't responding a lot, and we needed enough of them to leave the center that the stats wouldn't make a
You
difference.
should see, if they have a response, you should see it much shorter.
Yes, when we did the lights, they weren't running for that long.
Okay, thanks very much, Alana. Well done. Yeah, and then you can. Okay, so we're moving on to the next presentation, and we're sticking with the theme of shrimp fisheries for the moment. So I'd like to welcome Francisco for your presentation and best of luck.
Well, my name is Francisco Flores. I was working in the same lab as my colleague Alana there. And well, I was working a very similar topic, but slightly different, as we will see. Well, this is a little fine. Shrimp fisheries, as my colleague said, are one of the main contributors to bycatch in the northern Baltic Sea. When targeting this species of shrimp, crangum. I took these two pictures in Belgium. That one is from Germany, but it's basically always the same. A lot of flatfish, a lot of cod, juveniles. So it's very important that we start looking at options, right? Because this is not very sustainable in the long term. So we focused on pots, as my colleague said. Pots are very good as an alternative because they are very versatile. You can fish on many different bottom types, sand, mud, rocky shores, or like Alana said, in between offshore wind farms. Also, it requires a lot less fuel. You can use smaller ships to deploy a lot of pots. If the weather is bad, you can just go the following day and the shrimp are still going to be alive. And most importantly, there's a lot less bycatch. And this bycatch is still alive when you retrieve the pot, so you can release it back into the sea. But the main disadvantage from pots is that they don't catch enough shrimp, as we were seeing before. So we started looking at options, at possibilities, of all the possible ways to make pods more efficient. And this is when I decided to study light as a way of making these pods more efficient. What do we know about light being used to attract shrimps into pods? There have been some small trials in the past in different places, but for my research, these were the three main foundings. Humberstad in 2018 deployed some pods at sea targeting pod using lights of different colors, and he observed that cod was not being attracted to the pods because of the light, but because of krill that gathered around the light, and then that's what attracted the cod. So they did the same year some other experiments in the lab, and they confirmed that krill showed this phototaxi. Very similarly, the same year, Baumeister in Sweden did his master thesis in something very similar, deploying pods with light targeting the shrimp Pandalus borealis, and he observed that indeed you can increase the catch exponentially combining light and bait. But he suggested that maybe it was a similar mechanism than with the cod. Maybe the shrimp were not really attracted to the light, but just being attracted to something smaller that was attracted to the light, like larvae or some other organisms. But most importantly, what inspired this master thesis is this CIMAPA project last year, carried out by the same laboratory where I was working in. They were targeting Palaemon serratus using these light-equipped pods, and they accidentally caught a lot of crangon. So this, when I arrived in the lab, and we asked ourselves this question: can we really attract crangon using light? As Alana showed in her presentation, we had these 500-liter tanks with 4 compartments, one central compartment where we had 50 shrimps initially, and I decided to study white, green, and blue light. We put the shrimps in the middle, we let them for an hour to acclimatize, and then we removed the cork stoppers that were blocking the exits, and we let them for 5 hours, and we counted how many were in each compartment. This leaves us with a very simple model, just the cod shrimps in each compartment in function of the treatment, which is the light types, the compartment, just to see if there was some unaccounted variable like the water flow or the position in the lab, something that was making one of the compartments catch more shrimps than the other, and a random factor just to account for the effect of replication. But additionally here I wondered, what if they are not being just attracted to the light, but they are just trying to escape the high density of individuals in the middle? Because this was something like this big, and we're putting 50 shrimp, so maybe they were just trying to get out of there and not really being attracted to the light. So I ran another few replicates using only 10 shrimp. That's why here I had to add an offset variable with the initial number of shrimp, because I also wanted to use those for the main experiment, right? And then I also continued with these questions. What else could be biasing the experiment? And I asked myself, what if shrimp are, again, not being really attracted to the light, and it's just the impression, and what they are doing is trying to escape or explore the tank, and the only exits they can see are the ones with light. Maybe they're not really going towards the light, they're just are the only exits they can see. So I had these secondary setups. In this one, I put the lights in the center, so if they are really attracted to the light, they should just stay there. And this second one, I just removed all the small lights we had and just turned the lights in the lab on to make sure there was light everywhere, and if they wanted to explore the tank or just escape from the center, they could totally do it. And for this model, also very, very simple, what I did was comparing the total number of shrimp leaving the center with the ones I had observed in the main experiment. and the results were very satisfying. All the compartments equipped with light consistently caught more shrimp than the control, as you can see, and the green light caught significantly more shrimp than the white, but all of them performed a lot better than the controls. But not only this, we also saw that only when the Pisces was the name of the lights we used, only when the Pisces lights were put in the lateral compartments, in the main experiment, that's the only occasion when the shrimp really left the central compartment. When we left the lights in the center or just the general lights on, shrimp just stayed in the center and didn't choose to leave. And this difference in color performance is very worth studying, mostly because seeing that they all perform good could be used for anti-bycatch devices. If we see that they all work, maybe we can study which of them attracts less bycatch and then try to figure which is more sustainable. But this is not everything. We still have a lot of work to do. As my colleague said, trials at sea are the logical next step. There is no way of recreating all the possible variables in the lab, and if the project got more funding, we would probably do it. Also exploring different pod designs. It's been seen in other pods for other species, then the smallest change in design can really increase the catch a lot. And of course, explore other attractants like artificial smell, or even sound, or anything else. Or even different types of light. I wanted to try UV light or red light, but we didn't have time or resources. And here I wanted to just show this 20-second video of this, the funnel coming out of the central compartment. And this was one of the lights, and you can see the shrimp going directly towards the green light. In one of the, it was just one of the replicates. It wasn't always this good. Sometimes it took longer, but this time I was very happy I recorded this one. Thank you very much.
Thanks. So there's a couple of questions already, so audience, get
Thanks for your presentation. Just wanted to know, so you find different intensity of shrimp or the different light color, abundance of shrimp for different light color. Did you find this? They were spreading into the blue, green, and white. It was significant.
Yeah, green light had significantly a better performance than white light, and blue was somewhere in between. It wasn't significantly different from either of them.
And my question was, did you measure the intensity of light to see if there was maybe not color but intensity?
Yeah, it was written in the plans, as many other things we didn't test, because each replicate took a whole day, and then many of them we couldn't use because there was some small problem, like one of the lights was off, or one of them ran out of battery, or an endless amount of problems. So we had to reduce the things we were going to test. But yeah, that should definitely also be tested.
Thanks. Another question?
Very good presentation. Do you know why the green was more significant? Is there a biological reason?
Yes, more or less. The reason behind each of the colors, well, this is the one my lab tried and accidentally caught the crangon, so we had to try it. The Bowmeister, the Swedish guy who did his master thesis in something similar, he saw that white light was the best light for that species of shrimp, and other papers studying the spectral sensitivity, not for crangon, but for a very similar species, crangon almani. He saw that the spectral sensitivity peaked at the green wavelengths. So we decided to try, hoping that it would be the best, and indeed it was. So it was very satisfactory.
Thanks. Yeah, maybe one quick comment.
Thanks for your presentation, it was really good. This might be a little bit like of a reach, but one of the things that you're going to do next, hopefully, is to test this in the wild, right, in the open ocean. Do you all expect there to be like a difference in efficiency between what you tested in the lab and the ocean? Because my understanding is right now the tank sits in the center and have nowhere to go but towards the light. But in the open ocean, it's like a vast space and it's going to move towards a very minute, like very small specific area. So it's very hard to predict what will happen in the wild. The problem also with light is that it doesn't reach very far. So for fish, for example, that move a lot, like cod, you can use light as an attractant because it will pass near the pod and then see it. But shrimp that maybe don't tend to move that much, if you deploy the pod and then you just catch the shrimp and run it in. So it's very hard to predict. But the good thing about blue and green wavelengths is that it travels further than a red or orange. So yeah, it should also follow the same pattern ideally, but it's very hard to predict.
Okay, thanks. So we'll move to the jury. Who wants to start?
Who's this lady? Thanks very much for your presentation. It was very enthusiastic and well done. Just a quick question. So in the beginning you mentioned that this was kind of accidental, that we realized that they were, from the paper that you showed, that they were attracted to the light when you're trying to look at cod as well. Now, is that what happened?
Yeah, well, the second paper, it was nothing to do. It was the first paper.
The first paper, yeah that's right.
The first paper, they were targeting cod. They just wanted to increase pod efficiency for cod. And they observed with underwater cameras that it was that first the krill gathered around the light and then the cod came after the krill. So it wasn't a direct attraction. So something also very good about, well, very good. Something something I think we did right was using filtered water, so we didn't have these smaller organisms that could maybe attract the shrimp indirectly. It was only water and shrimp, and they still went after the light, so we can say a bit more.
But how would you prevent then the juvenile fish from going for the shrimp now? Because the whole thing with the fisheries is that we end up getting all this white fish as bycatch, and that's the biggest problem of the shrimp fisheries. So how do I still, like if I use light, how do I still prevent getting all this bycatch, which is the goal, I suppose? Yeah,
the thing is, each species responds in a different way to different light. For example, for cod or for flatfish, it's been seen that some lights can actually repel them instead of attracting them. So as here we saw all of them perform quite okay for shrimp. Maybe we can see which of them repels the bycatch, and then we can combine these two pieces of information.
But if they see a lot of shrimp, isn't that what they
right.
would be attracted?
You're right. Yeah, that would be. Well, the good thing is that the entrance for shrimp pods is quite small, so a cod is not going to.
But a juvenile, which is what not.
It might happen. Yeah.
So we still have the same problem.
But,
probably smaller, I suppose.
Yes, smaller and like a small flatfish.
Smaller
minus,
fishes. Okay, thanks very much.
Well, nice experiment. I have some doubts about the way you analyze the data. Can I ask you to put the model? In the first slide we have the model.
Here.
Okay, so you did not do all the data treated at the same time in the same model?
Not with these setups.
So
it
this
was a
model
different.
is just to compare the density?
This is just to compare the total catch between these two setups and this one. The total catch, not by compartments. Just adding the.
But you had some with the lights outside in each compartment, low and high density.
So, sorry, come again?
You had high density and low density with the same setup, with the lights on the side, no?
When I wanted to use less shrimp, 10 shrimp instead of 50 to compare the initial density, yes. For that I only use this setup.
So you compared only in that model, you compared low density, high density.
Yes, I compared the total proportion of shrimp leaving the central compartment when I was using 10 shrimp and when I was using 50 shrimp.
Okay, and you use light, the
For that,
type of light was in the model. How many replicates did you have?
with 10 shrimp, I had 3 replicates, and with 50 shrimp, I had 9 valid replicates. I did
Okay.
twice as many, but I
And you
could.
changed the position of the colors of the lights?
Yes, randomly, every replicate
Okay.
was in a different
All right.
position.
Okay, so then when you looked at the lights in the center and in the outside, you compared only the same density with lights inside and the outside.
For here, when putting the lights inside, I compared the total number of shrimp leaving, so the addition of these 4 compartments between the 3 different setups, these two, and the other one.
And those are all green lights?
Yes, because I was already seeing that it was the best performing light, so I decided to just have all of them green to make
Okay.
it more. Yeah.
Can you go back?
Of course.
So, when you put in the light type is where there?
The treatment.
Treatment.
Yes.
Okay. When you did that, you did like, for that particular treatment, you considered the 4 at the same time. The significance should have come from the control against the other 3. How did you compare blue and green and green and white?
Well, we treated each of the compartments as a different replicate,
Yeah,
and then we did that.
compartment goes in 2 places. 1, 2, 3, 4, and green, blue,
Yes.
white control.
Yes.
Okay.
Yes.
So when you test the light, you are in the treatment factor,
Yes.
the type of light.
Yes.
When it is significant,
Yes.
what it tells you is
Yeah.
at least one of them is different from the others.
Yes. Well, yes.
Okay.
Now I understand the question.
So
Yes.
I understand this is significant
Yes.
because you had one control that was very different from the
Yes.
others. All right.
Mhm.
How did you test
Yes.
that green was better than blue and better than white? Going back to a question you asked, are they significantly different?
Yes.
How did you do that?
We run an analysis with the function. It means we run after having the model done. We had basically an ANOVA comparing the 3 different lights, and it showed
It showed what?
it showed that
So you had an ANOVA comparing these 3 lights
yes, and the control. All of them were obviously, as you said.
for the high density setup.
No, the high density we only compared the for the difference in when using different initial densities, we just compared the addition of all 4 compartments here,
Ah, okay.
and that was just a correlation test. Is it the same proportion leaving when we changed the density? And yes, it was always around 75% of the shrimp leaving, independently of the initial number of shrimp. And yes.
Okay. What you presented is a factor in a multi-factor model.
Mhm.
Okay, so, my point is, go back to the
Yes.
slide of the 3 colors. I don't see, no, the colors.
Here.
That one. I understand when you put this in the model it's significant, because we have a control that is very low. I don't see there significant differences between the 3 groups. And I would like to know which test did you use to compare blue and green, green and white, blue and white, that tells you they're significantly different.
Yeah. It was a comparison of means using an ANOVA after having the model done. And indeed, blue wasn't significantly different from any, only from the control. But green was significantly different from white. That was the only.
So your assumption that green is better is absolutely arbitrary because you don't have information of
Yes.
statistical difference that green is better.
Yes.
Yes?
No, I mean, we know it's better than the
Not-
white,
not from
but
the
we don't
blue.
know if it's better than blue.
Okay, so when you have a
Yes.
non-significant difference,
Yes.
you cannot go ahead and interpret it is significant even if you have higher value.
Absolutely.
Okay,
Yes.
it's
Sorry if I
So
expressed
be
it
very
that way.
careful
Yeah.
because that is a serious mistake in interpreting statistical data. What you know is that light attracts the shrimp. You need a lot more information and more experiments to be sure that
Yes,
green is better than blue. And
Yes.
all right. clarify that.
You're right. Yes,
That all
absolutely.
your assumption that green is better is in your mind, it's not in the data.
Yeah. Okay. Thank you.
That's it.
My turn, okay. This is because the old fisherman believes that the green one is the good one. Isn't it? Isn't it? I mean,
The assumption. Well, we have the assumption. This assumption, looking at the spectral sensitivity, using the.
no, because when I go fishing,
Yeah.
I mean locally, all the people choose the green one.
Yeah, because it's the.
For the sweets or for whatever.
Yeah, the bioluminescence tends to be green, so people. Yeah.
So just on this some conditions, you have to say that the green is there. But I agree with the terms of intensity. I think it should be really important to see the intensity.
Yes.
And together with that, this is the device, the device you use.
It's called Thesis light from Safety Net technology. It's mostly being used attached to other nets to prevent bycatch.
You know how it works, I mean.
It's just yeah.
It's a battery.
Yeah, it has a battery, but we can recharge it wirelessly. So we just put it like in a suitcase, it charges, and then we can use it for like several days.
Okay, so
It's waterproof.
from the sustainable point of view, it's really nice to use batteries in the environment, and probably you are going to lose the batteries and everything.
Yeah, that's actually a good question. Ideally, like the thing with these lights is they're also very, very expensive. So for now, it's not really a good option for fishermen. And if they do use it, I'm sure they're not going to lose any of them because it's really something very expensive. But yeah, if they are used in hopefully a near future, that should be considered, and yeah, batteries should be limited or
I think that's
yeah,
a good
absolutely.
idea to consider just not to lose any batteries.
Yeah.
Of course. And I know that they are already losing because I told you that there's a lot of fishermen that they go with lights, just two squids,
Yeah.
whatever, and the lights are in the bottom of the
Yeah.
of
Yeah,
the
yeah.
sea for sure. For instance, my brother.
Yeah, but like even if some batteries could be lost, it could be a matter of considering what's the least bad option.
You believe that this is something for fisheries?
Yes.
It should be a huge amount of batteries.
Yes, yes, yes. That's right.
Just to consider when we talk about the sustainability, sometimes there could be other problems as well that should be taken into consideration at least. And I think it's
Thank you.
Thank you.
Thank you. Well done. I'm afraid to touch it. That's why I look at you. Okay, so we're on to our final presentation in this session, moving far away from shrimp fisheries to blacktip fruit sharks, and I welcome Lucy for your presentation, so good luck.
Thank you all for being here, and congratulations to my two colleagues for their great presentations before me. So as we just say, we're going to move on to a bit bigger animal with the blacktip sharks that I studied in Galapagos at the Charles Darwin Foundation. I know some of the jury was there during the presentation of my colleague Mar, and she already did a great job setting things up, but I will try to still give you a bit of context about sharks and the Galapagos Marine Reserve. So, 37% of elasmobranch species that are raised in sharks and chimeras are at risk of extinction according to the IUCN Red List. And this risk is mainly due to fisheries, but also additional pressures such as pollution, habitat degradation, and climate change and ocean acidification. In the marine reserve, the sharks and elasmobranchs overall are also facing the same pressures. And in addition to their high ecological relevance as top predators and top down regulators of their ecosystem, they also have a huge economical importance for the local communities, especially because of tourism. So why is it important and why are we doing wildlife health assessments? Well, first because it gives indication on the individual levels about pathologies and the state of the animals, but it also advises on the population health and state overall, which is particularly interesting in marine reserves such as the Galapagos for viral diseases and such. And it gives indication on perturbation over time. So for example, climate change, pollution, or fisheries can create stress for the organism, and that can be observed through physiological parameters. So the main way to do wildlife health assessments is through blood analysis. It's one of the less invasive ways that get the more results. Obviously, it's still invasive at some level, so there's always a little stress bias in the results. But it's a good way of getting lots of information from hematology and blood chemistry, which I will detail a bit later in my presentation. What is known so far in, black-tip sharks are the species I work with, Carcharinus limbatus. They're ranked vulnerable in the IUCN Red List, and they are one of the 3 main species with scalloped hammerheads and Galapagos sharks in the marine reserve. They are distributed all over the marine reserve, and that's why they're also really interesting to study for health and physiology, because they really reflect the state of their environment and can be considered as bioindicator species. So what is known so far in Galapagos about shark physiology? Well, not much. There's this one study by Laraga it's actually her thesis in 2021, where she looked at stress in juvenile black tips in San Cristobal Island, just here. And she looked at 3 parameters that are glucose, lactate, and hemoglobin, depending on stress. Other than that, there's nothing in the Galapagos. And in the world, there are some studies, but baseline assessments and assessments of parameters such as hematological parameters are really scarce, and there's really a need to enhance that database so that later on we will be able to assess the long-term perturbation I was talking about before. So the aims and objectives were then to assess individual health through blood chemistry and hematology, but also to set a baseline for the black-tip sharks in Galapagos, and for this study to be used as a pilot study for long-term health monitoring in sharks of the Galapagos Marine Reserve. I had 3 hypotheses getting into these studies. The first one was that hematology should be similar as other black-tip sharks population in the world and other shark species, as it is something that does not vary a lot. I was expecting fight time, which is the time between the shark bites the bait and is secured onto the side of the boat, and handling time, which I define as the time between the shark bites the bait and is released and free swimming into the ocean again, to be correlated with stress indicators, especially lactate, glucose, and pH. And my last hypothesis was that the stress levels should be lower than in other studies using more stressful methods. In general, commercial fisheries that use long gliders are very much a very intense stress for the animals. So how did I achieve that? Well, I did my sampling at the south of Santa Cruz Island in the Galapagos archipelago. We took this small fisherman boat with which we sampled the sharks, so by handline capture, and I was working with this beautiful animal, which is also always really great to be experienced. And we did blood sampling through caudal venipuncture, so once the shark was put in tonic immobility on the side of the boat, it's a bit of a sleeping state, and then we were able to do blood sampling. And we also took other parameters such as blood, length, sex, and mainly visual examination of any external characteristics such as bacterial infections or wounds that could be reflected by the state of the internal parameters. So from the blood syringe, we did biochemistry on board. We did slides on board as well, and then I stored the blood in 3 different types of anticoagulants that are heparin, citrate tubes, and in 10% formalin to do different types of analysis. From the 10% formalin tubes, I realized total counts of red blood cells and white blood cells. And with the heparin tubes, I did another set of slides in the lab because in the boat moves a lot, so it's not the easiest thing to do. And through the slides, I was looking at differential white blood cell counts. So I was looking at all the differential types of white blood cells that you can have to have both total numbers and specific numbers for each white blood cell types. For the blood chemistry, I use this device, which is called an e-stat, and it works, it's really magic. You insert a card with a drop of blood in it, and it just reads all the parameters that you decided before. So the card is called a cartridge. I use the CG4 Plus and the Chemic Plus to have a different set of parameters. So, what did I get? So we were lucky enough to get 12 sharks in just one field trip, which is really good numbers. They were all female, which is really great for me as for physiology, sex matters a lot. And they were all adults, and the fight time was about 7 minutes, plus or minus 2 minutes. That will be information that we'll be using and enhancing a bit later. So for the CG4 Plus cartridge, we got results for 10 sharks. It didn't work for 2 of them. And for the Chemate cartridge, we didn't got any results as sharks were most of the values were outside of the detection range. So this big scary table are all of my results for both hematology and biochemistry, but I will walk you through the main results and discuss them right now, because otherwise there's, I think, too much to say. The first main result I want to talk about is hematocrit. Hematocrit was found at 27%. What you can see on this graph here is the result that I got here in red, and then references in other black tip population with standard deviations. Every time it's the mean that's indicated. So to compare to the same species around the world. And you can see that I'm pretty much in the average, so results seem to not be alarming at all. Why is it interesting to look at hematocrit? Because it can indicate some pathologies such as anemia. Anemia is described in fish as when hematocrit is inferior to 20%. We did have one shark that was at 18%, so we could say that it is slightly anemic. However, in some shark species, it's normal to have slightly lower levels than 20%, so I would be very careful with that interpretation. When looking at red blood cells, we also had really good numbers of total cells, and the cells were looking absolutely great. So that's important because when you look at the morphology of the cell, whether it is the color, the shape, or the nuclei, it can indicate some pathologies, disease, and especially contamination. For example, if you have, so this is from another study, where you can see some abnormalities at the nuclei, and if you have micronuclei such as here, it can indicate, for example, contamination by heavy metals or other DNA damage. However, when I was looking at my total white blood cells counts, so white blood cells are really interesting because they reflect the state of the immune system and the immune response. I had very low numbers of white blood cells counts when we compared to, so this study did an average of 11 species of elasmobranchs, but basically every number that I found for white blood cell counts in elasmobranchs were more around 20, 30, when I have, well, 7.2, so quite a big discrepancy. Why is that? Lower blood cell counts, white blood cell counts can be due to stress, but stress can also cause them to increase. It can be due to also maturity stage or the reproductive cycle. I think that in my case it was most likely an error of observational bias, as I was using the 20x magnification on the microscope when I should have been using the 40x, as is most protocols about this method. Why did I not use the 40x? Well, it was broken. There was no other microscope available at the time I was in Galapagos. So I did my best, but I cannot really use all the potential from this analysis. Here you can see the differential counts for my study in the first column, then another study on black tip sharks, and then on other sharks. So as I said before, it's still very scarce knowledge about hematology in sharks, so I couldn't find much more references. It's basically respecting my results are along the lines of results from other studies. You can maybe wonder why my lymphocyte percentage is closer to the lymphocyte percentage of sandbar sharks and so far away from the lymphocyte percentage in other black tip sharks. Well, if you look at these two studies right here, you can see that they also have a big difference, even though they're in the same species. That is because in hematology and blood parameters overall, there's a very strong intraspecific and interspecific variation, especially in sharks. So what matters the most with these results is the ratio of each cell types compared to each other, and that ratio was globally respected in my results. So I can validate my first hypothesis that I would have similar results to other sharks. However, if we go to the correlation that I did with the Pearson correlation test, I got significant correlation for base excess, pH, carbon dioxide, lactate, which are all influencing each other and all in relation to each other, so that was expected. However, I did not find any parameters correlating with fight time or with handling time. What could explain this lack of correlation? Because that type of correlation has been proven in many other species and many other studies. Well, I think it's first I was working with a very small sample size of only 12 sharks, and even less because the carriage and biochemistry did not work in every shark, and even less because some samples were bad and were outliers that I took off. So in the end, I was really working with just data from 8 sharks. And also, as I told you before, the fight time was of 7 minutes with a standard deviation of 2 minutes. So it does not show a wide range of time that could allow me to assess a wide range of response in the physiology. And if we look at the stress parameters a bit more in detail, so if we look at lactates, so same concept, my study is here in red, all the other studies are in black tip populations, so same species but in other parts of the world. And I have quite similar results, especially when we compare fight times and lactate concentration to other studies, and you can also see that my sharks had lower lactate level than sharks from longline fisheries, which is kind of what I was expecting, and so that was expected. But if we look at pH, so once again my study is here, in pink are the longliners, and they're all about black tip sharks. So when we compare time, once again, for same fight time, the pH was about similar. However, it's a bit unexpected that I would have such low level of pH that are more indicators of stress. So that could be because pH is influenced by both lactate and carbon dioxide. Lactate was not showing any sign of stress, but pH was, so I guess that the influence of carbon dioxide was higher for the pH. It can vary depending on species. And pH is the main driver for post-release mortality. So what is showed here is that there was some physiological disturbance. However, when I compare the pH values to the other studies, they're always relating to sharks that were surviving and that were not showing any mortality after the capture event, even from satellite tracking. So, third hypothesis, I could say that lactate was for me a very good stress indicators, but blood gases absolutely not. And for pH and bicarbonates, it was higher than expected, more stress than expected, but still in the range of sharks surviving the capture event. And I will conclude briefly. So that's just what I was saying, that we had some physiological disturbance. It's always expecting when working with wildlife and doing some kind of even a little minimal invasive techniques. However, it had no implication on survival, and from my result I can say that it was less stressful for the animals than longline fisheries that are widely used in commercial fishing. And lactate was one of my most solid indicators of stress. I successfully described hematology for the first time in black sea sharks of Galapagos, and I hope that this study will be used indeed as a pilot study for future assessments in the Marine Reserve. I had to face a lot of challenging, especially regarding wildlife monitoring, all the variations I was talking about, and once again, the major flaw was my sample size that was really small. But overall, it should have implication and hopefully show that black tip shark could be a nice bioindicator species and very relevant to monitor for conservation and management decision. Thank you very much.
Well done, very good. So over to the audience first, if you have any questions for Lucy. Yeah.
I just don't know what is the difference of the long line and your
So longliners, they have basically it's the boat, and there's this line behind with hooks hanging, and so with baits, and then it's mainly for fish like tuna and all, but most of the time there's bycatch of sharks. And so the thing is that they drag this line for hours. It can sometimes go to, I don't know, like 100 hours of dragging the line. And so if the shark bites at the beginning, it can stay on the line soaking and fighting for 7 hours, 8 hours more. And of course that causes stress and freeze. What I did was use a headline that my supervisor threw in the water with a bait and some buoy attached. I'm trying to go back to my slides. It's a bit hard. So then we had a very reduced fight time in comparison to long line. So here you can see. So we had the boat, we just had a hand line, and then we had a buoy. So it's mainly the time that changes, but also because the stress generated by my study was more acute on a short time, while on longliners it's still acute on the time scale, but it's on a very much longer time. And we also, like, the shark were kept in the water to stay at minimal stress levels. And there was this buoy that we used. So the buoy is here, and the shark is somewhere over here. And the buoy was helping with the vertical traction, and my supervisor was doing the horizontal traction. And yes, they were trying to keep the fight at the minimum.
Great presentation, very interesting. I kind of have two questions. The first one, I don't understand any physiology or hematology at all, so I'm not sure if it's helpful. But I want to know, with your second and third hypothesis, how are you sort of differentiating the stress that is caused by the fishing or the long line or whatever the method away from all of the other stress that the shark might be feeling on a normal day?
Yes, so that's also one of the flaw of this study, I guess, because it's impossible to have controls in that kind of situation. So what I did was mainly compared with literature, because that's also the only thing I could really do. And when comparing with literature, what was really interesting was to... oh no, okay. Sorry. Okay, so here, for example, for pH. So when not all the articles specified fight time, but when I was comparing fight time, like in the city, like with Nernol, for the same fight time, I had about the same pH. So it really shows that there's a correlation to time, even though I was not able to assess it. Another thing that I would say is important for stress level is in some studies they did get the sharks out of the water on the deck of the board, and they did observe that it was higher stress for them to be hauled out in the water, maybe exposed a bit to the sun and to air a lot. But indeed it's really difficult. And that's what I was saying at the beginning, that there's always going to be some stress in studies with wildlife because it's impossible to completely shield the animals for stress. Another way that I can say that my sharks were not too stressed is that most of the time comparing my results were very close to other minimally stressful approaches, such as line fishing, and every time it was higher than drumline, it was less stressed than for drum lines or for longliners.
Okay, I think my question was more like, for instance, assuming you saw stress, how could you conclude that the stress was because of the way you fished, or rather than the fact that maybe the ocean was really warm, or there was predator effects, or any other kind of stresses that is actually impacting the physiology and not the fishing method?
So I don't think that's really possible. Regarding water temperature, most of the studies that I use in my comparisons and in my reference, I think almost all of them were at the same temperature, so seawater temperatures around 26.5 degrees Celsius. So that's a bias that I took out. But of course, life history of the sharks and reproductive stage and all of that will influence a lot of the parameters and the stress, and also that the way they react, because there is something that's really interesting, that there's always this notion of fight time in all of the studies, but there's never quantification of the fights. And when I was on the field, I observed that some sharks were were moving a lot more, even on a short period of time, and some sharks were not moving too much but still taking more time. And so that's something that is also probably very important, especially for blood gases and respiratory exhaustion and from stress. But yeah, there's too many parameters that could really go into. This was just to try and show that some methods can be minimally stressful to get more of a baseline data in comparison to methods that could have huge bias like longliners. Does that answer it?
So to the jury members, questions?
Okay. Well, thank you. This is not at all my field, so I'm going to ask some questions.
Yes, please go.
The correlations you showed, we couldn't see what they were. The letters were very small, but I couldn't understand what the lines were, what they were doing there. What are the lines?
The lines are just trend lines of my data.
You said they're not significant correlations.
No, these ones were the significant one because the p-value every time was very small. However, they were just not the ones I was expecting. I'm going to say, so this was lactate and pH with a p-value of 0.04.
Yeah. The p-values are there, you cannot see.
Yeah, okay. So, sorry, I know it's a bit small. So carbon dioxide and pH p-value 0.009. Correlation between base excess and pH p-value of 0.0005 and base excess in lactate 0.002. And that were the only correlations I had that were significant.
Those are significant?
Yeah.
Oh, it's good enough, no? It's very good. You expected other things to be significant?
Yeah, well, because these are all factors that all influence each other, especially lactate and carbon dioxide that are acid and bases will obviously have an impact on pH, that is a measure of the acidity, and on base excess. What I was really expecting was to have correlation with flight time and handling time, and actually any of the parameters, but mostly with lactate and pH. And I think because of my times were all so close to each other, and also my sample size very small, I was not able to really see a correlation with time and the increase of the response. So yeah, that was results that are significant, so yes, good in a way, but expected, and the ones that I was expecting and kind of counting on were not there.
So your comparison of, I think hemoglobin, the one that had the bars with several washers, well, that one is good enough. What are the bars there?
Standard deviation.
So if you have a 95 confidence limit, you basically double that. Not in this one, but the other one, there's no differences with the different authors. So interpreting which one is... That one, if you put twice the size of the bars, the means are all inside the bars of the other trials, meaning there's no differences there. You should interpret this as one being higher than the other.
That's something that I could not represent on the graph, but from other studies that didn't have the standard deviation of exactly the mean, they do mention in several papers that the normal pH level for sharks is between, I think, 6.8 and 7.
The mean, you mean? The mean level.
Yeah.
The normal is the range.
Yes.
So it is no differences there. No point in going and saying one is higher than the other.
Yeah.
Okay. Now, my other question is about the handling of the shark. I can imagine situations where you put less stress on the shark, or where you do not pull the shark to the vessel, where you take the vessel to the shark. Since it's hold on a line, you shorten the line by making the vessel closer. You quickly take a blood sample instead, maybe with an harpoon or something that doesn't require tying up the shark, moving into the vessel. Can you imagine a much easier way of collecting the sample, even if you have to give up some information on the visual evaluation of the condition of the shark?
Actually, blood samples in sharks are quite difficult to get. And this was, so the person that you see sampling the blood is a qualified veterinarian. It was one of my supervisors in Agneto Clodin. And it sometimes took several takes and several syringes before being able to really get it, because sharks, it's not like humans, you cannot see the vein through the skin. So it's really tricky to find, and depending on the position exactly and the type, like the morphology of the shark. And also there's another trick with sharks is that the venous, the artery is like this in the emerald arc. and there's the vein and the artery that are super close, and we were trying to take only mainly venous blood. So that's something that I cannot imagine being done through harpooning the animal. Yeah, absolutely not. But maybe there are more less stressful methods to approach the sharks and get them. That's the best thing we could come up with the means, the financial means that we had, and the localization and everything. But for blood sampling, it's really has really to be done in close proximity.
All right. So my last question is, from your research, what would you consider important for future studies?
That's a good question. I think for me the main relevance of this research is to be able to set some baselines that will later on be able to let us be able to compare with other differences. There has been a lot of trials in laboratory on how ocean acidification and climate change would impact shark physiology, and they found that it was causing some disturbances in acid base, that it was causing some changes in the distribution of the white blood cells, and even sometimes in the number of red blood cells. So that's something that's very interesting to know. And for me also the main point is pollution, because pollution is something we can really have strong action on by forbidding some pesticides or some the use of some products if we know that there is a high risk of runoff into the sea from agriculture, for example. So yes, I would say that long-term monitoring on health would give very good insights on the shark population state, but also on the environment and the contamination. Sharks are bioindicator species as they accumulate this kind of changes through the food chain. So if the species below them are contaminated with mercury or with other pesticides, it will show in the sharks. And that's why it's really interesting. And also because sharks, even if they are physiologically disturbed by this kind of capture, they're still quite strong animals on which we can, black tips are more than hammerheads. Hammerheads would be really disturbed by this. But it's still animals on which we can conduct this kind of experiments. And it's really insightful for them as well.
Tolerant to wrestling.
Yeah.
Okay.
Yeah. Hi, Lucy.
Hi. How are you doing?
Let's get into the hematology.
Yes.
Yes.
Cell biology. Wow, really nice presentation and very nice experiment, and together with that, the permission needed to do things like that in Galapagos.
Yeah, complicated. I was working with the Charles Darwin Foundation, but the authorizations are given by the national park, so there's a lot of administrative work to do beforehand that was handled by my supervisor, Gabriel Bayana, to get the authorization. And for example, for this project, we got the authorization to get blood samples and measurements, but we did not get any authorization for biopsies. So for example, this, I would have loved to get a biopsy of this. That would have been super interesting, but we were not able to because we didn't have the authorization, and it's actually something that's very highly monitored and under surveillance in the Galapagos.
Good, and it's important to be conscious of that because sometimes it's not that clear, even for the administration.
Yeah.
We can have some experience with our administration that they don't really know if it's possible or how to do things. So it's important to take into account.
And that's why also I was supposed to have 2 more field trips, but every time got cancelled, and then it was cancelled for logistic reasons, for weather, and because the fisherman coming with us was hurt himself. But then after to report the field trip was really difficult on the administrative level.
It's important to understand that these permissions also give guarantee to the spelling. So it's not only also because it's someone that is support that is looking for everything is going. Biochemical analysis. And that's the nice machine that you say something like it's a miracle. No, it's no.
It's that.
Okay. Do you believe?
Well, I think so. It was just I used it first because one of my advisor, the vet has a lot of experience with it, and also because it actually is in several studies about sharks. It's used, so I found many papers using the CG4 plus cartridge, not using the chemate. But then after, when I was doing my discussion, I did find one paper that said that the chemate was, for the chemate, the blood had to be diluted. But because sadly I found it after, I was not able to do this dilution, and then all of my values were out of range.
Make any kind of repetitions?
We did some, but not too many.
And they were consistent?
Yes, we did 2 replicas that were consistent. We did not do more because it's very expensive.
Yeah,
And we had only, I think, 12 or 15 cartridge of each type. So initially we were supposed to do 3 field trips, and we caught 12 sharks just with the first. So yeah, we could not.
Some experience with not these similar ones.
Which one?
I cannot remember.
Because I know there's also the vet scan and
I
some.
cannot remember because finally we discussed it because it was an item to work with. So I will ask just the reference later on.
Yes, I do.
Because it was really impossible to obtain a really nice repeatability measurements with other animals, with other fish and other.
But this one is also, I've seen it also be used in fish, but once again, mainly the CG4 plus cartridge, because it's also not made for fish initially, so that's why the chemate failed. But it's, I think, depending on the cartridge, you can have really good and reliable.
Finally, we go to typical spectrophometer, but in a lab, not in a lab. So
yeah,
it's perfect. You made a comparison between sharks. In the blood cells analysis, or was. Do you think that the different sharks has a species specificity in amount of cells,
for white blood cells?
proportions of white?
I think from what I've read, it can vary, especially between, well, if you compare elasmobranchs to other fishes. Within elasmobranchs, not so much, but I think if, for example, an individual is really injured or has a really important disease, and that was mainly, so that's why I was mainly looking for individual values and measures, but because I did not have anything unusual with individual, I just sum it up as an average for my 8 sharks.
But depending on also the biology of the animal could be different in the amount of, because they have the immune system more.
Yes.
Whatever, I mean, it could be as well. I don't know if it's possible or not, but that's why also the data is so important.
That's
Because
like
it has another, but I can say that all these data is so important because it's so difficult to obtain.
Yeah. Yes. Yes, and that's a bit what I was answering to my colleague earlier, is that it's, so white blood cells, it indicates a lot, but there's so much variation between species and even between individuals, because life history influences a lot. If it was sick before, if it's all the different stage of life cycles. So
Maybe because they are trying always to compare between species, but maybe it's not need. I mean, it's more, we need more information about the species, because we have our medical issues are completely well here, but we are one species. No?
yeah. The thing is that for black tip sharks, I only found one study that was describing. So that's the importance of my work, is to be the first to describe hematology.
You are going to publish it?
Yes, we're working on an article right now. I will send you the link.
Please. Thank you.
Okay.
Okay, so that wraps up this session. We have a half an hour coffee break. Just to finish the session, I'd like to thank all 3 speakers again. So one last round of applause.
Are we offline or are we already?
We're online.
We are already. It's good to know. We are online. Everything we say is on the word of the internet. Here's the camera, right? Okay. Good afternoon. Good afternoon, online audience. Good afternoon, dear students, dear jury members. It is my honor and my pleasure to be here chairing the last session of today's thesis presentation. It's not over yet. Tomorrow morning we have more. I will already make an announcement to you for the students. Spread the news. When you are celebrating if you presented today, if you are in the dorms, please be conscious and respectful to your colleagues that are still presenting tomorrow. Okay? Thank you. So maybe to the online audience, as I believe most of you know who I am, I hope. I'm Ana Monteiro, the main coordinator of the International Master in Marine Biological Resources, IMBRC. And here with the jury, we have Professor Jona Mari Gomes, who is the local coordinator of University of the Basque Country, and representing partner University of Bergen in Norway is our dear Audrey Geffen. Is that the correct pronunciation? Audrey, also Valentin Hesling is going to be part of the jury as well as myself. That being said, each one of the presenters has 15 minutes for questions, as you should know and be prepared of, followed by 15 minutes of presentation. Remember, dear presenters, nobody knows more about your research than you. You are the main authors of your papers. We want to hear your story. It's my pleasure to call to the stage Zoe Mulder, who's going to talk to us about assessing population structure, health status, and parasite Bonamia ostrea prevalence in native oyster, Ostrea edulis, beds of Western Ireland for restoration efforts. Zoe, the floor is yours.
Thank you, Alana, for that introduction. I was wondering if I could just try the swiping first to see. Okay. And with videos, if I swipe, it'll play the video in the slide. Oh, you should click it. Okay, sounds good. Okay, now that's good. So, as Luana said, hello, my name is Zoe Mulder, and I'm very excited to talk to you today about my thesis, where I assess the population structure, health status, and parasite Bonamia austriae prevalence in native oyster beds off Western Ireland for restoration efforts. Starting off, I will begin by introducing the main character, the European flat oyster, otherwise known as the native oyster Ostrea edulis. This is a bivalve mollusk that can live greater than 20 years, and sexual maturity usually occurs around 3 years. The spawning season is in the spring or summer, where eventually larvae are released, and they preferentially settle on other adult and juvenile oysters, where the spat fuse their shells to hard substrates, forming oyster reefs. And you can see the different life stages highlighted in orange boxes. You have spat, juveniles, and adults that can go greater than 200 millimeters. Moving on to the distribution, their distribution or biogeographic range is in green in the map, and they live from the lower intertidal to around 50 meters deep. Next, moving on to ecosystem services. So native oysters are filter feeders, and therefore they can increase the water clarity and quality, and which can benefit other marine ecosystems like seagrass meadows. They also play a crucial role in biogeochemical cycles such as denitrification, as well as stabilize sediments and prevent coastal erosion. And their complex reef structures that they form provide shelter and food for a diversity of species, which increases biodiversity, and also provides feeding and nursery grounds for species of commercial and conservation interest. However, native oysters are facing numerous threats and stressors. They are functionally and ecologically extinct through much of their historical range, largely due to overfishing, habitat damage, pollution, invasive species, as well as pathogens. And this affects larvae and broodstock. However, I will focus on pathogens. Which then brings me to introduce our culprit, Bonamia austriae, which is a haplosporidian protozoan parasite that causes the disease bonamiosis in native oysters. This is a cytological slide of a heart smear of Ostrea edulis,
And basically, the B. austriae microcells, which are seen in the small purple dots, they attack the hemocytes of oysters, and this infection can spread to other tissues in the gill, mantle, and gut, causing death of the oyster if infection is severe. And these big purple blobs, are the hemocytes. And it has been reported that there's greater than 90% mortality of oysters once introduced into a population, in some populations, and the parasite can be transmitted vertically from adults to larvae, as well as horizontally through the water column, likely by filtration activities of the oysters. So native oyster populations in the UK and Ireland have decreased by a whopping 95%. And as a result of these declines, there are numerous conservation designations for the native oyster and a great need for restoration. Which then brings me to our project for my research questions and objectives. So I wanted to collect baseline information of native oyster reefs in Western Ireland that are remnant beds to inform restoration efforts. To do this, I collected data on population structure using size frequencies, recruitment of spat, condition index, which measures the health or the physiological state that the oysters are in, as well as the parasite B. austriae prevalence. Which brings me to our study sites. So these are the two bays in Western Ireland that our oyster beds were located in. So the oyster beds are denoted by red diamonds, and the two bays were Bertrabui and Kilkieran Bay. We studied one oyster bed in Bertrabui, which is labeled BER, and the rest of the oyster beds were in Kilkieran Bay, which all the stations started with an S for station. And our study was the first to assess B. austriae prevalence in Bertrabui Bay and across a longitudinal gradient in Kilkieran Bay. And here are just some photos of Bertrabui Bay, what it looks like above and below the water. Moving on to sample collection, 414 oysters were collected in Kilkieran Bay in December 2023 by dredging transects. And 118 oysters were collected in Bertrabui Bay by scuba diving in January and February 2024 using 50 by 50 centimeter quadrats. And here's me being very happy with a big bag of oysters on fieldwork. Moving on to the morphological analysis, we brought the oysters back to the lab, cleaned them as best we could of epifauna, declumped them, and then we measured the height and the length using digital calipers, and then categorized the oysters into life stage, into spat, juveniles, and adults based off of shell height. Here are just some photos of some of our samples. Moving on to the molecular analysis for Bonemia austriae detection. First, we took a subsample of oysters from each oyster bed that were chosen for B. austriae detection as well as condition index. And then the fun started where I shucked and took 25 milligrams of gill tissue from 162 oysters, and my arms and wrists were very sore after that. And then we lysed the gill tissue with proteinase K to break it up and get rid of the protein. We performed DNA extractions with QIAGEN robots and checked the DNA extraction quality and quantity on nanodrops. Moving on. Then I had many Eppendorfs of DNA extractions that needed diluting and prepped for PCR, wherein I did two standard polymerase chain reaction assays. The first being standard PCR amplification of fragments of B. austriae 18S rDNA, which then led to gel electrophoresis. And basically the oyster tissue samples that had clearly visible bright bands on the gels were counted as positive for B. austriae infection. And the real-time PCR amplification we did was of fragments of B. austriae actin genes. Each sample was tested in triplicate, which then, from this, we obtained threshold cycle values, which is basically the cycle in the PCR amplification where the fluorescent intensity increases above a background level because we're using probes. And then we calculated the average CT values for each sample, and those values that were less than 38 counted as positive, again, for B. austriae infection. Moving on, lastly, we did a condition index where we separated the tissues and the shells from each oyster, put them in an oven at 60 degrees for 48 hours, and then we measured the dry weights. And the condition index is simply the dry tissue weight divided by the dry shell weight times 100. Moving on to results. So I'm going to start with the population structure analysis. On the left here are size frequency histograms of shell height. Shell height and shell length were very similar because oysters have isometric growth, so I'll only focus on shell height. And these are for each of the different oyster beds, with Bertrabui up in the top left and Kilkieran for all the rest of them. And on the right, the two histograms, the top one is all of the Kilkieran Bay stations lumped together, again for shell height, and the bottom again is Bertrabui Bay. And just some things to note, As you can see in Kilkieran Bay, there was much more spat recruitment compared to Birchibouy Bay, lots of small oysters. But in Birchibouy Bay, we had the largest oysters in our study, where around 14% of oysters in Birchibouy were greater than the commercial size of 75 millimeters, whereas only 3% of oysters in Kilkieran were greater than commercial size. Looking at recruitment in more detail, so the oyster beds or stations are on the x-axis, and this is just the count of oysters. Yellow are spat, and navy are juveniles and adults. And the numbers denote the relative frequencies of spat. So what we can see is that there was a significant difference in spat recruitment among oyster beds. Kilkieran stations S63 followed by S28 and then S51 had the highest relative frequencies of spat. Birchibouy and S78 had low relative frequencies, and then the rest of the oyster beds in Kilkieran had no spat. Next, looking at bulimia ostrei prevalence. So this is a map of Kilkieran Bay with Birchibouy off to the side. Again, you can see where the different oyster beds are. And these pie charts denote the B. ostrei prevalence. So orange is the percentage of oysters in that oyster bed that did not have the parasite detected, and blue is the percentage of oysters that did have the parasite present. As you can see, Birchibouy, there's no B. ostrei detected, and Kilkieran, there was higher prevalences of B. ostrei in these oyster beds in the west, S63, S28, and S25, with greater than 20% prevalence, and then lower or no B. ostrei prevalence in the oyster beds in the east. And there were significant differences among oyster beds in the B. ostrei prevalence. Lastly, we did a multivariate analysis, multivariate principal component analysis, and this is just a PCA biplot of PC1 and PC2, and we used the response variables, presence, absence of bulimia, size, height, or which is height and length, as well as condition index, and then the groups are the location of oyster beds. I have labeled each of these quadrants to try and figure out the patterns a little bit better on the graph. And something to note is that the Birchibouy oysters, which are in red circles, were larger, or they grouped differently than the rest of the Kilkieran oysters, and they were larger as well as not infected with the parasite. And you can also see the variances explained, or the variation explained in our data by the principal components. And then this is a PCA biplot of PC2 and PC3. And again, I've labeled each of the quadrants, and again, you can see the Birchibouy oysters are grouping differently than the Kilkieran oysters. And again, they were not infected, but higher in condition than Kilkieran. And these samples up here were the oyster beds that had bulimia present, which were in Kilkieran. So take-home message is that the Birchibouy oysters were larger, they had a higher condition index, and had no infection with B. ostrei compared to the Kilkieran oysters. Moving on to the discussion. So these graphs are simplifications from many studies I looked at in the literature. And what they found is that there are various environmental factors that affect B. ostrei prevalence. These include temperature, salinity, and food availability. And these environmental factors also affect condition index. And so we know that in Kilkieran Bay there are large fluctuations in temperature, salinity, and nutrient levels between the inner and outer parts of the bay, as well as among seasons. So this would be very interesting to study these environmental factors in Kilkieran Bay in relation to B. ostrei prevalence as well as condition index. And again, a simplification from the literature, but other studies also found that there is the highest B. ostrei prevalence in spring in larger oysters, and this decreased towards the autumn. This decrease coincided with spawning. So during spawning, oysters release large amounts of energy, and they have a lot of biological effort they're expending. And what these studies hypothesize is that the larger oysters are dying in the spring, and that is why the B. ostrei prevalence is decreasing towards autumn and then is only found in the small oysters. And a similar trend has been found with condition index, where the lowest condition index values are after spawning, so towards the later summer, and then there's higher condition indices or higher health of the oysters throughout the rest of the year. So how does this relate to our study? So Kilkieran Bay oysters had generally lower sizes than the Birchibouy Bay oysters, so we can possibly infer that there's a high mortality of the large oysters occurring in Kilkieran Bay, where B. ostrei was present, and not in Birchibouy Bay, where there was a lower, or there was no presence of B. ostrei, and had higher or larger oysters. Moving on to connectivity. So this is a screen recording from an oceanographic model of the Connemara region of the currents. And here's Bertrand and Kilkieran Bay. And as you can see, there's not much connectivity or currents connecting these two bays, potentially explaining why we have B. austriae prevalent in Kilkieran Bay, but not in Bertrand Bay. So we can infer that the oyster beds are within their own self-sustaining, self-seeding systems within their respective bays. Now zooming in on just Kilkieran Bay, again, currents with the hydrographic model. And then overlaying our B. austriae prevalence map, we can see that there's a lot of connectivity occurring between the oyster beds in the west that also had the highest prevalences of B. austriae, like S63, S28, and S25. And again, not much connectivity occurring between these oyster beds in the west and those in the east, that again had low or no prevalence of B. austriae. And this can also give insight into our spat recruitment results, where we did find the highest relative frequencies of spat in these oyster beds in the west, and these in the east either had very low or no spat in our samples. So some pretty interesting results when we look at connectivity on a spatial gradient. For implications and future studies, I'll start with implications. So our study provided crucial information for restoration efforts in this region, such as site selection for where to add culch, and as well as oyster translocations, where and where not to translocate oysters based off of the patterns we observed. This has also important implications for biosecurity measures. Clearly, there's not much connectivity occurring between the infected oyster beds and the non-infected, and especially between these two bays. So fishermen, restoration practitioners, researchers, recreational boaters need to be extremely careful to not transfer this disease and this parasite between these locations of infection and non-infection. As well, our study provided information for the potential aquaculture of bulimia-free Oedula stock from Bertrand, and this needs to be looked into in greater detail. However, future studies look at environmental factors in conjunction with the parameters that we examined, like temperature, salinity, and chlorophyll measurements in Kilkieran and Bertrand bays, as well as incorporate a larger temporal variation with seasonal monitoring. Larval and disease dispersal could also be studied in greater detail with hydrodynamics, as well as connectivity and transmission pathways. Genetic and phenotypic analyses should also be examined to determine if there is a distinct strain of oysters in Bertrand, and other methods for B. austriae detection should be looked at, such as histopathology and cytology, which can give us information on the location and intensities of infection, as well as non-destructive and non-lethal methods like eDNA and eRNA, as well as anesthetizing the oysters instead of killing them for gill tissue. In conclusion, our study was the first to test B. austriae presence in Bertrand and a spatial gradient in Kilkieran. We provided important baseline information for restoration and biosecurity. Bertrand had a distinct population structure and health status compared to Kilkieran. Oyster reefs are highly productive, biodiverse, such as these photos here, taken from oyster beds in our study, but also threatened. And now more than ever, there's a need to conserve and restore the ecosystems we depend on and the services they provide. Thank you.
Thank you, Zoe. Perfectly on time. Or just on time. Which was still perfect for the record. Now it's time for questions. Jonah?
Okay, so thank you. Congratulations for a very nice presentation. And for the very quick working in the lab. So
I was not fast in real time.
I don't have to see people like working in the lab. No, I really don't have many questions. I think it's very well done and very well planned and very well explained.
Thank you.
And maybe yes, and also because you have projected into future research needed, how to proceed after this study. As far as I know, one of the main environmental factors regarding bulimia distribution will be salinity, for instance. And if you look at the mapping, it is called Kilkieran Bay. And you have the two branches of the estuary. I was wondering whether you go to the map. Go to the map.
A map, okay. Which map exactly? Not this one.
This could be any of those.
Okay.
So then you have to the left part, you have the bulimia with prevalence, and then you have the other part is not. So my question is, It is more fresh water the right brand, and is more salty water the other round, the other side of the round.
Yeah. No, that's a good question. And that's also why I wish we could have collected more environmental factors like salinity and the temperature and chlorophyll measurements in our study, but fortunately that wasn't possible. But yeah, so basically from what I read about reports in Kilkieran is that there's many, especially in Kilkieran Bay, there are many rivers and streams and lakes that are all flowing into Kilkieran, especially from, it's kind of cut out up here, but especially from the north part, there's a big river that goes in. So you'd expect lower salinity there compared to here where the open ocean
And
is.
between the two parts there, the two
Between
areas?
these two, again, I don't have measurements of it, but all that I read in the literature was that both the east and the west parts of Kilkieran Bay had a lot of rivers that were flowing into both sides of the bay.
Okay.
So maybe it would be a matter of more like these oyster beds or ones that are closer to the land having lower salinities due to freshwater influence.
And I don't know the area. I suppose it's an intertidal area.
Yeah.
So which is the point at which tide reaches in each of these places?
So I don't remember off the top of my head, but in Kilkieran, I think the lowest depth was around, like it's quite shallow, 20
Okay, I
meters.
will forward.
Okay.
Is any place in one of those sites
Yes.
in which the oysters are uncovered for long
No.
time? They're always subtidal.
They're always subtidal in this
Always
area.
subtidal.
Yeah, for all these beds.
And can you identify that there is a different degree of turbidity in the water because it's more muddy one area or the other? Because these are factors that can affect the transmission. And maybe regarding the prevalence of tsunami.
Yeah.
Can you distinguish them?
I didn't do the field work.
I don't mean whether you measured. Whether based on the knowledge you may have about previous studies, the area and so on. Can you say something about that?
Turbidity, no. From also my knowledge on all of the reports I read about each of the bays, they didn't measure turbidity or that from what I could find. There were measurements for temperatures and salinity, but I couldn't find anything for turbidity in Kilkieran Bay.
Okay, and my second and last question, I don't know. This prevalence is based on PCR analysis, basically.
Yeah.
So, in order to make, you want to do some, not prevalence like the incidence, the degree of infestation that you have in the oysters. You should do some microscopic analysis and so on. You have said it would be adequate to follow this way and so on.
Oh, for
But I wonder whether when you do the PCR, you detect that there is some different signal between different sites that may be reflecting that the intensity of infection is different in different localities or not.
Did I find that with PCR?
Yes, by the PCR.
No, so PCR only gives information on if the parasite was present in the oyster. So that's why I also mentioned, using cytology or histological methods where you're using slides and also sections, then you can actually determine the intensities of infection. Like the cytological slide that I showed earlier, you could clearly see that there was a lot of bulimia microcells in that slide, and that was a level 4 infection. That was the highest level of infection, and that's why I think this work should really be done. And like the gold standard recommended by the EU is using cytology, histology, as well as PCR, because PCR is more sensitive of a technique, but we don't get the information of the intensities or the locations of where the infection is in the oyster. So that should be followed up. We got our oysters frozen, and for cytology and histology, you need them to be alive and fresh. So it just wasn't possible with our study, even though that actually kind of was the original plan.
And did you get samples for histology?
No. But the Marine Institute.
This is a waste of something. When you got the animal,
sorry,
that is for histology.
I meant these not for my study, but yes, histological samples were taken by the Marine Institute and examined by the Marine Institute, because the Marine Institute takes samples for cytology, histology, as well as PCR, qPCR. And actually my qPCRs I performed at the Marine Institute, and all of my samples in Kilkieran were collected by the Marine Institute. Okay, so they have, they did take a lot of those samples for,
Okay, that's the final comment. You are using condition index as the ratio between the dry weight of the tissue weight and the dry weight of the cell.
Yeah.
And I know this is sometimes used in oyster farming as an index of calf condition index, but it's quite inaccurate because the cell weight, the cell is very fragile, so it breaks a lot, and then it is difficult to compare. And if you are looking for some physiological endpoint, it's much better to relate the weight of the meat, of the flesh, against the volume of the cavity that you can calculate with plastilin and the Archimedes principle into water.
Right, yeah. I did read a few papers that kind of compared all the different ways to measure the condition index, and now recently, all of the studies I found from 2010 use this as a metric for condition index, which is why I also used it, just because it's then comparable among studies. I didn't go into it in detail here, but in my manuscript, I talk about comparison of condition indices because they use the same methods in the other Cs and everything.
I know
yeah.
but you have breaks in the cells, or you have a lot of noise and background, so it's not very...
Yeah, and even it's hard to remove all of the epifauna, which then affects the weight, which then affects condition. So there's for sure limitations with it, but I just went with the most recently used method right now.
Okay, so thank you. Congratulations.
Thank you.
Thank you, Jonna. Audrey?
Thanks very much. I enjoyed a lot of the things that you were talking about, and different ways that you've done things and the different aspects that you've looked at, and such. I have a couple quick questions and some longer ones. But the first is a really quick one. Did you look at any seasonal effects?
No, I would have loved to. That would have been cool.
So I missed the time frame in which all of your samples were collected.
The oysters in Kilkieran were collected in December 2023, and Bertrabui January and February 2024. So really all in winter, basically.
Okay.
It's just that the samples in Kilkieran were collected before I arrived in Ireland and were frozen for me to use. And that's why there's also different sampling methods. They use dredging, whereas we use scuba diving to collect ours because it's less destructive, obviously. And then I arrived, I think, mid-January. So then we did the field work for Bertrabui, which is why there's a little bit of a disconnect, but they were all in winter.
And did you control at all, or could you control at all for density? Have some, extent of oyster beds or some kind of population and size index?
Yeah, we did look into that. Again, with Kilkieran, not so much because even the transect lengths varied from 50 to 100 meters. So we couldn't really get a good measure of density. With Bertrabui, it would have been a bit more possible because we removed all of the oysters from the 50 by 50 centimeter quadrats, then giving us an area, and then obviously with the number of oysters, so we could try and figure out the density based off of that. But because we couldn't do that with Kilkieran, we focused on other metrics.
But it was a trawl, right? It was a trawl, so you know the width of the trawl and you know the length, so that gives you an area. Yes, although I couldn't obtain that data from the Marine Institute. I need to... I didn't obtain it, no. But I'm gonna ask in the future. We're also planning to publish this, and they're obviously gonna be co-authors as well in the paper, so there's a lot more that we need to flesh out, but in such a limited time span, I was just trying to get my analysis on what I was focusing on done. Okay, one more question. You're proposing the connectivity is not really connectivity, but you're proposing that current is affecting the incoming of the parasite, and therefore those downstream are more likely to get it than those that are not in there. But how could you test it? So this is you're just sort of superimposing two different patterns. Yeah. But how would you actually test it? How could you test it? Test the connectivity. So one method that was actually proposed, and I think is already a good method because it's a lot less destructive and invasive, would be using eDNA and eRNA. So because this parasite can be transmitted within the currents, as well as in larvae that are traveling between the oyster beds, it has been, many studies recommend using either eDNA or eRNA. It really needs to be developed for the species and everything, because then you could really see where you're detecting this parasite along a gradient. But again, it needs to be much further. Has anybody done any drift modeling? No. There is actually, in the local masters in Ireland, there's a student that's going there in September, continuing on in this exact same project, and he is specifically looking at population genetics, but also looking into the hydrodynamics and going to be studying that in detail and likely doing drift modeling and everything. So that's a future study that's going to start in September. So very soon.
Thank you, Alden. Valentin, do you have any questions for Zoe?
Do you have time for?
Yes.
Yeah. Okay. Well, thanks, Nathan, for the
Not for
presentation.
the long, but
Yeah. Okay. I would like to go to slide 18 on your PCA.
Maybe I'll just, it'll probably be easier. Yeah.
Yeah, good. So why? I'm wondering where have you done PCA?
Why a PCA?
Because at the end, like your conclusions was like, yeah, we saw like I don't know size difference between the slides.
Yeah. So actually, I also did a lot of univariate analyses that I did not show. So I looked at just shell height in comparison among oyster beds, and we did a permanova. And condition index also, I did a permanova for, again, univariate analysis. But the PCA was just to really combine everything and get an overall visual of it. And then we also did a permanova on the principal components, PCs 1 to 3, because that standardized the data, kind of put them on a same scale. Because I'm working with binary data, continuous numerical data as well. So we did a permanova afterwards with post hoc tests as well. So there was more stats behind just this, but this was just to show it kind of all in a graph to get an idea of what was happening.
thank you. Thank you, Valentin. Do we have any quick questions from the audience? If not, I have one quick question. Just for the record, I give the 15 minutes for the questions. That's to be fair to everybody. Zoe, you mentioned already a lot of future things, the limitations of the work, and some projects that will start in September. So I'm not sure what is really for you the future plans, but let's say that you have limited resources and capacity to continue this project. What would be your planning?
It's a great question. I love that question. So I, first of all, would like to, it would be very cool to develop more eDNA and eRNA approaches, especially, again, less invasive. These oyster beds are already extremely at risk and threatened and are facing a lot of stressors. So this would, like our sampling is having an effect. So it would be a non-invasive, non-destructive approach to doing it. Probes, as I realized, for qPCR and molecular work are very expensive, so if I had a limited budget, that would be great. But also one thing, and we even talked about the local Gaeltacht oyster fishery co-op, is making Berkshire a marine sanctuary. It's currently not, but it is in such good condition, and it's one of the last oyster beds, as we know, in a lot of Europe that is in this condition, and it hasn't been fished. It has huge amounts of biodiversity. Again, within the same framework, there's a PhD student right now collecting data on the biodiversity that this oyster bed is supporting. And yeah, it would just really help research. It would help the fishermen to have a brood stock to relay and try and restore other oyster beds that they do commercially fish. So really to make that a marine sanctuary, that's what I would do.
Okay, thank you very much.
Yep.
Kiana. All right. Yes. Please come in. We are about to start.
Sorry, is that my first?
Yeah, I thought so. So no problem. Just in time. All right, so it's my pleasure to introduce the second speaker of this final session of the first day of Thesis presentations, Keanu Goodison, who's going to tell us about combating bacterial cold water disease in aquaculture, bacteriophage infectivity across time. Keanu, the floor is yours. When there's 1 minute left, I will stand up.
Right. Thank you very much. And yes, I will be continuing down the road of microbiology. and by looking at how we can use bacteriophages to treat a disease in cold water, in like bacterial cold water disease in agriculture. So first off, because we have a couple of people who are not marine scientists, I will start by saying, yes, agriculture is important, even in the European Union, where the industry is a little bit smaller than in other regions. It's still 20% of our fish products are coming from agriculture. And what we like to farm in Europe is rainbow trout, and that's also the case where I did my thesis in Denmark. And what these Danish fishermen, these Danish farmers are seeing is they're having significant problems with this disease, bacterial cold water disease. And that's because the principal, the cause of it, Bacterium psychophyllum, well, it's ubiquitous in the environment, which means you cannot keep it out of your farm. It will be there, and if you go to any Danish farm and you just take a random fish, they will have this bacteria inside of them, but it's not always causing an infection. So it's also kind of an opportunistic disease, is that it's just a matter of time before it actually starts causing a problem for your fish, starts infecting them and starts killing them off, especially if they are still young. And this is of course not what you want. So that's why we are looking to methods to treat them. So what you do normally is at the moment they use antibiotics to treat this disease. But of course we want to reduce our usage of antibiotics, and that's why I come present to you a phage therapy, because this uses bacteriophages. And now, if you don't know what a bacteriophage is, I will also refer them in this presentation as phages. But just remember, I am talking about bacteriophages. What they can do is they can infect bacteria. So they are sort of viruses that infect bacteria. And so also for the bacteria that is causing the disease, you can infect them with this virus. And what happens is, if you follow the lytic cycle, is that more particles of this virus are being formed until the cell dies. There is a cell lysis, so the bad bacteria is actually being killed. And more of these particles are being spread into the environment and can start infecting more bacteria. So one of the big uses of it is specificity. It can only infect a couple of strains of this specific bacterial of this specific bacteria, and it won't affect any other bacteria that might be healthy or beneficial for your gut, for instance. But that can also be kind of a problem because it might be too specific, because you only infect a couple of strains with those, that you actually have some strains that can survive and grow. So that's why in this thesis I want to isolate new phages, because we really want a broad range of bacteriophages that we can choose from to start constructing these cocktails that we can give to the fish to treat the disease. And also to identify which phage has the broadest host range, because those are the most promising ones that we can use. And also to see if there is a pattern in infectivity and how it changes over time. Can the phage infect the same amount of bacteria present in an agriculture farm this month as it can in the next month? And then I also worked a bit on optimizing DNA extraction, because initially I was going to work more on genomic data. So how do you isolate these phages? Well, the good thing is you only really need two things. Those are bacterial strains and water samples. And those water samples just come from the agriculture farm, and they already have a high abundance of phages, because phages are just everywhere. And everywhere where you have a certain bacteria, you will have those phages. We just need a method to get them out of the water and really concentrate them up to a very high concentration of phages, so you can start treating your fish with it. And what we use for that is called a double layer agar method. And there are two methods that are used a lot in the industry. That is direct spotting assay and direct plaque assay. And then one method that is being developed in the lab where I worked is using an enrichment culture, where you first give some time for the bacteria to multiply in a smaller volume together with the phages from the water sample, in the hopes that you have a lot more bacteria that can be infected by phage, so you end up with a lot more phages, and then you can isolate those phages from that. So then, what I went on to was an host range analysis. So how many bacterial strains can these phages infect? And that still relies on double layer agar methods, where you then, instead of spotting with the water, you have your pure phage stocks. So you have like one phage genome in one lysate. And then lastly, what I did for DNA extraction, what's important here is that I worked on different starting cultures, different starting volumes, to see whether this has an effect on how good the DNA extraction is, how much DNA can we extract, and how pure is the extracted DNA. And there is also a method that we started with to really give good opportunities to start extracting DNA from phages. So first for DNA results, you might see a lot of numbers here. What's most important to know is that in the 2 milliliter extraction, only 13 out of 32 samples had a DNA concentration that was higher than 2 nanograms per microliter. This is a lower limit for the company that we're working with to sequence the DNA. So we need to be above that 2 nanograms per microliter. So these are shown here. But then when you look at purity, so for those who know, it's measured by nanodrop, and it's the 260 over 280 for purity 1 and 260 over 230 for purity 2. And everywhere where you see an asterisk, the value that you expect is that you get is much lower than the value that you expect, which means that there are signs of contamination, which is again not good. And these are all the extracts that actually had enough DNA in them for sequencing, but they might get problems with the actual sequencing procedure because of contamination. Then we use 30 milliliters of starting culture. Actually, most of my extractions, there was way too much cell debris, so that was not very successful. Only 1 out of the 12 samples were DNA extracted. And that's why in the 10 milliliter, the ideal spot was found, where you can see everywhere that there are very high values for DNA concentration. They are all above that 2 nanograms per microliter lower limit that we need. And also the purity values, they are very much more in line with what we expect for a solution that is pure DNA. And yeah, here again, it's just more of the extracted, like with the 10 milliliter extraction. And then also for the phages, the method that we used, we were able to get high concentrations of DNA in the extract, and also good values for the purity. Now onto the phage isolation. As I mentioned, the 2 methods that are used a lot in science, in the direct spotting and the direct plaque assay, for me they were not so successful. With the 5 bacterial strains that I was using, actually only phages were isolated for 1 of those strains. The other ones we couldn't find phages. And yeah, those 2 phages were also purified. To a pure stock. Then for when we were using enrichment methods, we actually see a big increase in the potential for phage isolation. So here we see all the bacterial strains that we were using, and we were also using 6 different water samples that came from the agriculture farm. And what we see is that for each of these bacterial strains, we actually find phages in them. So we can isolate phages for every bacterial strain that was used during this sampling time, which will later be referred to the January bacterial strains. So yeah, this is a big improvement with what we found before, where only 2 phages were found. So this is just to give you a sense of how this looks like. And phages are quite easy to find because you have phages where no bacteria grows. So on this plate, everywhere where it's murky, you have bacteria growth, and this is like a nice bacterial lawn. Then here we have inhibition zones. That's because there are phages that are killing off the bacteria. So then when we go to the host strains analysis, we can group our samples, the strains that I used, into 3 different time periods. January strains, these are used also for the isolation. Then a month later, and then strains that were isolated before that. If you look at the global picture, we see that the phage that I isolated, FPS VD51, has the best infectivity ratio, which means it can infect 82% of all the bacterial strains that I used, which is a good start. And what you can also see, these are 6 older ones, the VD44 to VD49, and you can see that these values are a bit lower than for the new phages that I found. So now when we split all this data up into the 3 groups, again, these are the bacterial strains where we were isolating phages from. And you can see that, again, VD51 has the best potential of infecting bacteria. It can infect 70% of that group of bacterial strains, which is not surprising that it's good because we were isolating phages against those bacteria. But what's more special is that when we go back a month later, get more bacterial strains from the same farm, we actually see an increase in infectivity, and we actually have perfect infectivity for FPS VD51. It can infect all of the bacterial strains that were taken during that sampling time, or like isolated during that sampling time, which is very promising because that means that you have time to isolate phages, and the bacteria, they don't have in that short time a resistance to the phages that you isolate. But of course, we still have to see whether this transfers, whether this is like a one-time thing or whether this is the global thing we see in the whole dataset. And when we look at older strains that we were isolated way before, in 2018, we actually see some influence of when the phage was isolated on how many bacterial strains that they can infect. Because here, the older group, the VD44 to VD49, you have some higher effectivity ratios than with the newer group of phages that I isolated. But you can also notice a lot more similarities between them. You have a lot more same values, like 0.75 comes back 4 times across both old strains and new phages. So there is some influence of time on infectivity of phages. So I also want to share just the host range in table form, because here you see the bacteriophages, and here you see the bacterial strains. Everywhere where you have black color, you have infectivity of the phage on the bacterial strains. So the bacterial strain, or the growth of it, is being inhibited because it's being killed, in essence. But we can also see that, for instance, FBSD35, none of the phages used are able to inhibit growth for this one, which could be more problematic, especially if this is one of the more virulent strains, but at the moment we don't know that yet. But yeah, but if you remember back from the enrichment culture, there we could isolate phages for all of the strains from January, which means that there are phages that we can access, but just because of the ones I selected during this time in this thesis, they were not, although there is potential to still attack those bacterial strains. And what you also see here is we see some similarities between bacteriophages, but if you look at the other tables, you will start seeing some differences because each phage is different and has different infectivity pattern. It can infect different bacterial strains, and we also see this in the table. And there are no 2 phages that infect perfectly the same strains. There's always at least one strain that one phage cannot infect and the other one can. So these are the older strains. I also want to touch up on a little bit here that we didn't investigate fully. But here in this dataset, we also have some geographical differences from where the bacterial strains are from. So you have some Swedish bacterial strains and one Finnish one. And you can see that all of these are actually being infected by at least one, by at least one phage. So that's also very promising because we can isolate phages from one aquaculture farm and even go to a different country and start treatment with those bacteriophages in a different country. So here I try to again show you a bit of the similarities between the different phages. So in this group, you see a clustering of older bacteriophages. In this one, you see a clustering of newer bacteriophages. So that means that there is some importance on when the bacteriophages were isolated and their infective capacity on the bacterial strains. But we also see a group that has 2 newer bacteriophages and 1 older one. So that also means that there are other factors that influence bacteriophage infectivity. And they can actually have an important role because actually this VD50 is able to infect a couple of the bacterial strains that VD51 was not able to. And this I just want to shortly show because all of these 4 bacterial strains were taken from one fish. And you see this diversity in the bacterial community already. When you just look at infectivity by phages, you don't have this catch-all phage that can infect all 4 of them to a really good state. So that means that you really need those bacterial, you really need these cocktails of phages to use to really treat all of the strains that are present. So in short, just to summarize again, I was able to improve the DNA extraction by having that 10 milliliter extraction volume. I also showed that the enrichment procedure really increases your possibility of isolating phages, ones that we might have missed in the past. And from the host range, we see that that infectivity actually remains good in the short term. From January to February, we still have good infectivity, but maybe on the longer term, this dies off or this can still change. And then I also want to say that I would have really liked to work on more genomic data, and I think in the future that's also the plan of the place where I worked, to really include all of this into a genomic package and what is important. Sorry. That was, yeah, the questions.
Congratulations, Kenna, for this very clear and interesting presentation. Don't worry, it was just a little technical.
Okay, sorry.
No problem. Audrey, can we flip the order? Are you happy to start?
I don't have a lot of questions except to wonder whether phages compete. So the phages of different strains, when you test them, do you think they compete?
Well, you have a lot of cross evolution of bacteria and the phages together. So there's probably some, because you have this similar host range for some, and that can be an indication that there is some competition between them. But I think in a general sense, you would think more of the competition between bacteria and phages that keep evolving the phages as more of an important factor.
Thank you, Audrey. Valentin, do you have a question for
Yeah, thanks, Kino. It's a very cool presentation. I'm wondering, do you know what would be the limiting factor if you want to administer the bacteriophage to the fish directly? Like first, what would be the way to administer it? Would it be like a vaccine or through, I don't know, food or something else? And because I think the bacteriophage are quite sensitive to temperature and change of or light, you know. So I would like to have your opinion on that.
Yeah, so what the group that I'm working with is they're working both on food, so having coating it on pellets, but also actually on coating the filter, like the sedatives that you use in the biofilters, because you are working in an agricultural facility, and especially in Denmark, those are mostly recirculating agricultural systems. So there's a very high control in water quality there and in different factors such as temperature. And that means that they are very interested in having these coated just on like these plastic sedatives that they have in the biofilter, and adding them to the filter directly and leaving them there, and like every once in a while maybe changing them out for new ones with different phages to have a long-term suppression of the bacteria.
Okay, I time for a second question?
Maybe, yeah.
Okay, I guess this research is made to avoid the use of antibiotics that we know have a lot of negative effect on the environment. Are you aware of any potential negative effect of the bacteriophage to the environment?
And that's the good thing about them. They're so specific to Flagobacterium that, because we isolate them with this method, that you really select for specific phages that only affect Flagobacterium, only a couple of strains, that you don't have a lot of negative effects to it. At least not that I'm aware of.
Thank you.
Good. Okay, thank you, Valentin. Jonna?
Yes, thank you. So thank you for the presentation. It's been very detailed. I learned a little bit of something I don't know so much, so it was quite appealing in a sense. I have some question that maybe is related with what Valentin was saying is, if you compare not only the effectivity that they may have when you apply against antibiotics, because it's not clear which is the way of application. You have said that could be capsules or water or whatever. I don't know to which extent that the phage need to be activated or kept there in conditions, how expensive this could be. So you put it into economic terms. How much will be in favor of the phage instead of the antibiotics from your point of view?
It would actually be in favor of the phage because they are quite cheap to produce. You only need the media to your bacteria growing, and then like some starting phage. And when you add them to the bacteria, that's like the ones that you use to isolate them against, where they have very good infectivity in the phage, then the phage, out of just a couple, like a day, you'll have so many phages that you can just use that as for like many different applications. So that actually makes it a very cheap method once you have your phage stock. Because you can just have big of those bioreactors where you produce the phage. And then, so yeah, it's like a lot of research that I read say that it will potentially become cheaper than antibiotics.
Okay, just one curiosity. Is the disease in the fish is related to cold waters?
Yeah.
So basically, the risk exists in the winter months. So you don't need to apply the fake treatment during the periods of potential risk for the disease, not the rest of the time. However, if I understand it well, the cold winter disease is associated to a combination of the immune depression in the fish together with the activation in the lactobacterium. And that's what happened probably is that at the time the bacteria is in the lag phase of growth or whatever, and it's just the time in which they are less sensitive to incorporate phage into the cells. So it would be possible to use more in a, maybe it's stupid, but in a more preventive way. Yes, when the bacteria are in the steady state phase, be exposed to the phage and not wait until this the break out of the disease when you have the phage lag. Could you some sense on that?
That's why they are looking at both the biofilter application and the coating on the food. So the coating on the food is really like the fish are sick and we want to treat them. But the biofilter, that is really meant as a long-term, like passive suppression of the Flavibacterium, where you just constantly have phages in your system and like relatively high quantities that can suppress the growth of the bacteria, and then once you start developing, because eventually they will develop resistance to it, then you want to change to a new phage cocktail on your biofilters, like change some of the cell tips to new ones with new phages, and then you can move, like really extend that suppression of the Flavibacterium. But of course, it's still under a lot of development, so there are still a lot of tests that you have to run before really there.
I'm talking about the concept.
The concept they are looking at. So.
But this is why I would be worried about the long-term treatment, because you were saying that okay, it's very specific for these particular strains and so on, but it's specific under these conditions. But if you have the fakes in conditions for this higher temperature, their host can change, the infectivity and so on can change. So it's not any worries that they will be affecting other bacteria which are not necessarily the pathogen ones, just because you are making an overload of these fakes in the system at high temperatures, for instance, if you are applying it all around the year. Is there any risk of general infectivity escaping from that?
That's why it's so interesting to work in Denmark, because they have these recirculating agriculture systems, and they can really control water temperature there. And they have said that in the summer it's a bit more difficult, and that's also why they have a little bit more outbreaks in the summer, because cost of cooking the water to 15 degrees the whole time can be expensive. But their facility is made to maintain water temperature at a pretty stable level.
Thank you. Congratulations for myself.
Thank you, Jana. Do we have any questions from the audience? If not, I have a question. Okay, there in this facility, I can just imagine an amazing infrastructure, but that's not the case, as you probably know, around the world for aquaculture systems. Do you or your collaborators are thinking on how do you track the mutation rate of the bacteria to see the effectiveness in the longer term of the bacteria phase?
They are busy with effectivity experiments, so that's where you are measuring them with different equipment to see how the suppression happens over time, and like you have these really nice curves of growth suppression, and then at the end you can actually see differences in the bacterial bacteria that you take out of there at the end. So for instance, what is linked to these phages is that there might be a loss in what is called gliding motility. That's more of a morphological difference between the bacteria. That they start, well, gliding motility is that they can sort of move a little bit across like your air plate, that they don't remain in a perfect spot, but you have like multiple spots of where the bacteria is growing. And that's, for instance, something that changes when you are infecting them for over a long time with bacteriophages. So yeah, they are looking at that, and that's also why adding genomic data to it can be really important to see are there genomic shifts there or not, and is there just selective pressure that we are adding there that are causing, well, not causing, but that result in some genomic shifts or not. So.
Okay, thank you very much. Is that, yeah, quick question?
I don't know if it's relevant or not, but have you seen any difference depending on what the organism that is affected on? Have you seen any difference, or have you even looked at different species that could be that are affected by this?
Well, there are multiple species affected by that bacteria, and especially when you look at the whole family of bacteria, it's like a very common disease. But because in Denmark you pretty much form rainbow chart and not a lot else, it's also the focus of the whole research group and of the whole partners in the project.
Do you think there would be difference, or what do you think?
Well, of course, you're dealing with different systems. You might not have the same bacteria represented there. So yeah, there might be a difference. It's not the focus of the group because we are so busy in rainbow charts.
Thank you very much.
Thank you for your question. Thank you, Kevin, for an excellent presentation. While Valentin is setting up the presentation on screen for our next presenter, the last but not least important, of course, just an announcement for the partners and the students. There will be a group picture taken outside immediately after the end of today's session. So don't run away, because we would like to have everybody on that group picture. Of course, we will have other pictures taken on Friday, but we want to take pictures here on this side. All right, so it's my pleasure to introduce you to Zainab Farsi, who is going to tell us about the impacts of ocean acidification and elevated alkalinity on the growth and survival of juvenile oysters. Zainab, the floor is yours.
Hello, everyone. I'm very pleased to introduce you to my topic, which is about impacts of ocean acidification and elevated alkalinity on the growth and survival of juvenile oysters. So, in recent years, with the rapid development of industrialization, a lot of anthropogenic activities has caused the level of CO2 to increase in the atmosphere. The CO2 doesn't just stay in the atmosphere, and it is absorbed by the ocean, which leads to what we call oceanification. So since the 1950s, the atmospheric CO2 has been rising steadily, and as a consequence, the dissolved CO2 has also been rising in the seawater, which caused the pH to decrease and make the water more acidic. So as a consequence, the pH of the ocean, the surface water, has dropped down from 8.16 to 8.07 since pre-industrial times, which means a 26% increase in acidity. So looking ahead from now on, if this trend continues on, we may be even looking into 100 or 150%, 100% increase in acidity.
to explain a little bit more what happens exactly when CO2 enters the water. It interacts with with water and form carbonic acid, which quickly dissolve into bicarbonate ions and can also turn into carbonate ions and hydrogen ions. With the increase of hydrogen ions, of course, the water is more acidic, but this equilibrium, with the increase of hydrogen ions, this equilibrium can shift more into the favor of bicarbonate ions. So they tend to form more bicarbonate ions instead of carbonate ions, which is a threat to marine bivalves or shellfish in general, especially in their early life stages, because they use carbonate ions to form their shells. So instead of forming their strong shells, they tend to have deformed shells that are easy to dissolve. That is also due to the carbonate saturation states, which are affected in these conditions. The carbonate states are basically the ratio of the carbonate ions and the calcium ions by the product of solubility product constant. And when this saturation state is above 1, the water is supersaturated with the carbonate we want, and marine calcifiers have it easier to form their shells. What we wanted to look at basically with this study is to search for more solutions to maybe mitigate this the effect of ocean acidification in aquaculture studies. First of all, this is where our study has taken place. It's the Rio Formosa lagoon in southern Portugal, and it is known for bivalve farming due to its rich waters. This region specifically the Itma Tavira, where our research has been conducted, a lot of ocean acidification research have been carried out, but what was interesting is that most of them found neutral or positive effects of this ocean acidification on the bivalve. Their conclusion was that maybe due to the local high total alkalinity in the station, of the seawater of the station, that could have maybe buffered the effects of ocean acidification on the bivalves. So we tried to test this. So our first experiment was to see the effect of 2 different total alkalinity levels on the growth and survival of 2 different species, Pacific oyster and European flat oysters. We were expecting to see that maybe based on the past studies that elevated total alkalinity may enhance the growth of these oyster species. Our second experiment and most important one is to assess the effect of elevated CO2, but with raised of the oyster juveniles, specifically for the Pacific oysters, but when raised under elevated total alkalinity conditions. And we were expecting, of course, negative impact, but hoping to see that if that total alkalinity could maybe mitigate these effects. So first of all, our biological material for the Ostrea edulis or European flat oysters, it was sourced from the Ipma Tavira, from the local broodstock. They were 7 months old, around 20 millimeters in shell length. For the Meaganaticas or the Pacific oysters, it was taken from a nursery in Oliao, and they had the same age and around 12 millimeters in shell length. For the Ostrea edulis, they were already acclimated to high alkalinity conditions since they were raised in the Ipma. Contrary to the Meaganaticas that was raised in the nursery in the waters of Rio Formosa. So what we did, first of all, our first experiment was set up in a way to have each species had 2 treatments, one with the normal total alkalinity water and the other with the high total alkalinity water, with each having 4 replicates and 30 oyster individuals per experimental tank. We took the normal alkalinity water from the Rio Formosa lagoon, or the Thesis beach that was close to the station, and the high alkalinity water was straightly sourced from the station. And the levels of the total alkalinity were around 2,300 micromole per kilogram per normal alkalinity water, and 3,200 micromole per kilogram for the high alkalinity water. For the second experiment, we built a system in the station. First of all, we have the water that is directly sourced from the Rio Formosa lagoon, and it goes through a lot of steps of purification and treatment, and it goes directly into 2,000 liter tanks for aeration. This water is directed to two header tanks of 2,000 liters that are connected to pH stat or CO2 controller that we fixed at 2 CO2 levels for the control at around 8 pH level, and the other at around 730. And we tried to keep open water system so the water goes directly into the experimental tanks, where we had 50 individuals per tank. These tanks had a volume of 20 liter per experimental tank. Yes, and for the measurement and analysis, we took the seawater variables every morning. We also measured the total alkalinity and the carbonate system variables. We also took the biometry variables that we measured them at the beginning of the experiment, after 15 days, 30 days, and into 54 days of the experiment. We also measured the condition index. It was the same as Zoe did. We also had to measure the shell organic content. They were both at the start and the end of the experiment. Yes, so going into our results, first of all, for the Pacific oysters and their different total alkalinity levels. First of all, we have the shell length in the y-axis, and the x-axis has the different time points, with the normal alkalinity being in blue and high alkalinity in red. So the shell length for marine energy gas, which was, as we hypothesized, is that it had higher growth in high alkalinity compared to the normal alkalinity, except there was no significant differences, unfortunately. The width has a similar trend, with high alkalinity having a higher growth increment versus the normal alkalinity, also no significance. And lastly, the height also showed a higher difference, a higher growth in high alkalinity compared to normal alkalinity. So the takeaway message from this is that there was an overall better growth in high alkalinity, despite this species being already acclimated to normal alkalinity conditions. For Ostrea edulis, or the Pacific oyster, we found that after 54 days, they had much higher growth in high alkalinity compared to normal alkalinity. The same thing for width, but there was a significant growth at the very end of the experiment, after 45 days. As for the height, which was a little different than the others, it almost did not grow at all in normal alkalinity waters versus a much higher growth in high alkalinity waters. So for Ostrea edulis that was already acclimated to high alkalinity water, it suited the species much better. Okay, for the next measurements, we also took mortalities during the whole experiment, but they were not relevant. So, I mean, the title of my thesis is survival, so I had to include them just to make sure. And for the condition index, it showed a higher growth in in normal alkalinity, sorry, but there was no significance. The the shell organic content was also showed a higher growth in normal alkalinity waters versus high alkalinity waters. For Ostrea edulis, there was almost the same trend, but there wasn't that much of a difference, also not significant. So they had a better condition under high alkalinity waters compared to normal alkalinity waters, and this shell organic content was also higher in high alkalinity waters. Yes, so Ostrea edulis had a better condition index in high alkalinity waters, but the other species fared better in under normal alkalinity waters. For the second experiment, which involved the two CO2 levels and high TA, the shell length showed a higher growth in the high CO2 levels, which was what we were hoping for, and it was significantly different after the 45 days of the experiment. The width, however, showed a much higher growth in control conditions compared to the high conditions, and it really grew by a lot in those 45 days. The height was similar to the shell length, and it did grow much significantly higher than in high CO2 conditions compared to control conditions after 45 days. So we had a greater growth in shell length and height in high CO2 condition, and width had a higher growth in control condition, which was really interesting because despite the low saturation rate in the high CO2 conditions, we saw that two of the biometry variables had a higher growth in high CO2. For the mortality is the same. And yes, for the condition index, the control showed lower health or lower value than the high CO2 condition. So they had better health in high CO2, but unfortunately no significance. For the shell organic content, however, the control group was significantly higher than the high CO2 group. So they had a better shell content in the control CO2, but they still showed a better condition in the high CO2 conditions. So the key findings were that juveniles of Mayagana gigas and Ostrea edulis grew better under elevated TA conditions compared to normal TA conditions. But despite the high CO2 levels, Mayagana gigas still did grow better than elevated TA, which brings us to our objective that elevated TA conditions could potentially be a solution to mitigate the effects of oceanification on shellfish farming. But this is still just the beginning, so we should definitely investigate the long-term effects to elevated total alkalinity, also on different life stages, because it can change with the time. We can also study the combined effects of other environmental stressors such as temperature, salinity, also in the scope of ocean warming and all of that. And finally, since this could be a solution in aquaculture sector, we could also evaluate the economic viability of this solution in those operations. And it's thank you.
Thank you, Zaina, for another interesting presentation. Now it's time for questions. So, Valentine's, our time to start, please.
Yeah. Thanks a lot. It was well presented. I enjoyed your presentation. So congrats on that. I have one question. Don't you think, because you say that the oyster were taken from local farming? Yeah. Don't you think this population could be already adapted to this specific environment? So
yes.
Maybe that could explain. Yeah.
Yeah, but like for this specific species, Ostrea edulis, we only included it in the total alkalinity experiment, not the ozone acidification one. It wasn't a part of the plans at first, so we just wanted to see how different acclimatation histories could impact maybe the effects of this variable on how they adapt and how they grow there. Yeah, that was basically the point. At first we were just going to go for Pacific oysters. We took them from somewhere else, but once we went there, we saw that they had a small broodstock of this species, and we said, why not just try it and see what could happen.
Okay, good. And the second question, so you took the condition index,
yes.
the SOC and mortality.
Yes.
So as your, so that on your result that the, maybe those proxy were not very relevant for such a short experimental time?
Yes.
Well, I just want to have your, I don't know.
Yeah.
What's
but
your opinion
it
on
still
that?
gives us an idea on how these species are doing, regardless of the significant or not, because sometimes the oysters can deploy their energy resources into different axes of growing, and the actual condition index could be different. So maybe they could have higher growth than in high total alkalinity conditions, but you may find that the actual condition index is higher in control conditions. So it was mainly interesting to see how the show organic content was still higher in the control conditions, as the condition index was still higher in the high CH conditions.
Thank you.
Thank you, Valentin. Audrey?
Thank you very much. And it was nice to see the two species being used there in very different conditions and all. But yeah, it's been a long time since we've had, come back again to the ocean acidification work, which is quite nice. So, can you explain what you say mortalities were not relevant? Do you mean they weren't significant or they weren't relevant?
I think I have.
It's like
so these are the actual numbers for the mortalities. So, for example, in the total alkalinity experiment, we had only 2 mortalities per experiment. For example, for the Mayaganaji gas, so in all of that experiment,
So why is that not relevant?
Because it wasn't tied to any other. We couldn't explain it with anything else. Maybe it was just a competition for the space, or because we had smaller spaces, and it wasn't. It was mostly because of the time period. Because normally there is other studies where you compare the mortality to maybe like seasonal or temporal variations, but here it was more controlled conditions.
But these are averages in the different tanks, right? In alkalinity, right? Is that the average, or is that across all of your?
No, across all of the 50 days of the treatment.
You don't think it's? I mean, I think that's remarkably low mortality, and I would think that would be very relevant. Because it shows what a good experiment, everything was in good
I mean, if you see it from that side,
I'm asking you to tell me why you think it's right. Why it's
no,
not
I was
relevant.
thinking from the point of view that it didn't affect their conditions and all that. That's how I said it's not relevant.
Okay. But it means you have more. I mean, it's quite important.
I agree.
And I guess I'm also curious why you expected to have a negative effect from ocean acidification when the previous studies didn't find it.
Well, the previous studies did not specifically look at the. Some of them did have negative effects, but just like some of them did have neutral or positive effects, and they could only hypothesize that it could maybe due to the total alkalinity. So just taking the history of ocean acidification and all of the research done, we said that maybe normally it hinders the shell formation and all that, so it should be negative effects. So we were basically looking for the buffering effects of the total community.
Some calcifiers grow faster.
Yes. But I don't know.
Okay. Yes, Jana, please.
So thank you for the presentation. It was very interesting. And I wonder, you have quite complex experimental setups. Will you be taking part in the design, setting up, taking care of all these things? Because I know it's a lot of work.
Yes, it is. So for the design, it was based off what my supervisor did in her previous research for the design of the experiment. But for like the monitoring and all that, I did everything myself. I had to be there.
So you were doing the running of the experiment, you were there?
Yes, the whole time of the experiment I was there. I was doing all of the biometry, the measurements, taking all the seawater variables every day, and checking the mortalities every day and all that.
Okay, no, I say that because I find that to make such a complex experimental setup means that you need to train many diverse type of data. So I guess there will be more data beyond those you have presented on the biometry of the shell, let's say. So I guess that there will be other members of the team will be processing the soft tissues, looking at different endpoints like enzyme activities or composition or whatever. You have any data? Is already there?
For this step of the experiment, we mainly focused on the growth and the survival. The other type of data were just like the seawater variables and how they changed over the course of the experiment.
What did you do with the meat of the oysters?
We measured the condition index with it.
Only that?
Yes, only that.
I'm quite lazy and quite group research is very expensive. You have to do a very big investment in everything. So you need to be very effective in obtaining the maximum benefit possible from a scientific point of view of what you are doing and working as less as possible. This is two main principles of research. And so in this case you are doing a very huge work to make these experimental designs. I guess looking at the growth of the cells and the flesh of the meat, I think it's a tremendous effort for a small package.
We do have some of the oysters that were left freeze for maybe potential other.
Okay, so you have more samples
yes.
that can.
For now, this is where we stopped. We were hoping that maybe we could do more analysis on isotopes, or also on to see, for example, if because we also took the sea variables, we also freeze some of the waters we took because we had to take the water directly from the sea to see if if there was something weird or maybe something that like an outlier or anything. We also have the water freeze to check what could be different in the composition of water during that time also. So there is more to come.
Yeah, I said that because I was surprised because of the results. They were not significant. So
yes.
You have very good trends, so probably you need to have a thought to see, okay, it seems there is something, you can identify it probably because the statistical approach is not the correct one. Maybe you should try to modify parameters, transform them, look for correlations or whatever.
What we thought is that also the period is not enough because some of the graphs, they showed significance after a certain time. So maybe leaving them for a longer time could maybe give different trends. So
yeah, but the other issue is that you are to some extent focusing on cell growth, even if you are yes, cell growth. I mean, if I were a farmer of oysters, for me it's not the most relevant thing. I don't want to have big shell oysters. I want to have big meat oysters. And so, and indeed, it's a problem with some of the contaminants that they produce, thickening of the shell, and abnormal allometric growth of the shell, and so on. And this is not good. So you are taking as a positive that they are growing more, but depending on the energy budget, they can be growing more by cost at the expense of the meat.
That's true.
And this is not good for the farmer. And so you need to look at more endpoints in order to make these kind of statements, even if you cannot classify this thing. This was one of my main things. Also because you are combining two things which are important for cell growth and from growth in terms of growth rate and metabolism, which is alkalinity and CO2 concentration. And alkalinity will be affecting the anhydrase carbonic, which is the enzyme in charge of building the shell in mollusks. And the CO2 concentration at high levels will be producing hypercapnia, which is a physiological effect with the basic metabolism goes up and the growth goes down because you need to spend more just to stay. So the experiments are very nice, and I will get much more harvest for them. That's why I do it for the same price. It is important. So this is my general advice and reflection.
I mean, I agree with you because we also had a time constraint. We were supposed to start beginning of January, but we, due to materials not arriving until mid-March, we did not start that early. So I had to just hurry up and try to come up with something important. So this is mostly like the beginning. We try to see if we can validate the effect of total alkalinity, and after that we can move on from there.
Thank you. I don't repeat the experiment because the result will be different.
Thank you very much, Ivana, for relevant observations. Yes, Peter, we have time for a quick question.
Okay. If European and Pacific oysters seem to grow well under increased acidification, do you expect that they'll expand their range in an increased warming or climate change scenario, or do you think the negative effects will cancel that out?
Well, we did not actually test if the oyster, also Pacific oyster will grow better under high CO2 levels. It was only the total alkalinity experiment, so I'm not totally sure.
Okay, no worries. Thank you,
Thank you, Zainab. So I'm not going to ask questions now, but congratulations for your work. And thank you for explaining also the constraints that you faced while doing the experiment. So I think it's impressive that from March to now you were able to, and obviously you got good knowledge. Now time is up, so thank you again. Please, calm down. If they take the picture without me, I'm gonna be quite upset. So,
I'm taking the picture.
I really want, please, I really want to end this session by congratulating all the three speakers of